Национальный центр биотехнологий является одним из ведущих научных организаций биологической профиля в Казахстане. Основой нашего центра являются
Доброе утро, уважаемые гости и участники конференции.
Good morning, dear guests and participants to the conference. Welcome, dearest guests. We are glad to welcome each other, and let us give a round of applause. We're glad to greet you to the International Conference Astana Biotech 2024, which is devoted to the 30th anniversary of the National Center for Biotechnology. We would like to congratulate you with the achievements of the Republic of Kazakhstan in this area of biotechnology. We're going to be sharing and getting exposed to R&D innovations, and let us celebrate the 30th anniversary since the National Center for Biotechnology was established. And before we start, I would like to ask a question. Janar, we're going to be talking about science. However, do we know, what comes first, an egg or a chicken? I think that unfortunately we won't be able to find out why, as the truth is hidden in the threads of the discussion. So I think that we're going to be having a lot of discussions and disputes. There are many speakers to deliver their speeches, and I will ask you one more question. So why did you decide to join science? Science is magic. Scientists create new concepts. They get their theories developed. So therefore it's pure magic. Magic is wonderful. So let us start off starting the magic. And what are we going to be talking about? The conference is going to be divided into the following sections: biomedical technologies, technologies for agri-tech, section number 3, biotech for food processing industry, and section number 4, biological safety and security. And in the same information gets in Kazakh language. There are going to be lots of sections devoted to environmental protection, for instance, and we're going to be talking about the safety. And today we have invited prominent scientists from all around the world to include higher education institutions at neighboring countries, and our colleagues from abroad have also joined today's event. So in the end of the conference, we're going to be discussing a lot of questions to include genetics, and we have also invited the representatives of the prominent producers and companies who make equipment. So there's going to be a mini symposium, the diversity of genetics in Kazakhstan, and we're also going to be having the exhibition of equipment and services being provided by the leading companies. So it's high time for us to start. So let us start with the very beginning of how this has started, from those who were the pioneers in terms of science. So I would like to give the floor for introduction to first Vice Minister of Ministry of Healthcare, Mr. Sultan Ghazi. So please. So first Vice Minister of Healthcare, Mr. Sultan Ghazi. Please, the floor is yours. Good morning, dear colleagues, and good morning, representatives of the world of science, and on behalf of the Ministry of Healthcare and on behalf of myself, I would like to appreciate you for participating in the celebration of the 30th anniversary of the National Center for Biotechnology. Congratulations! And Astana Biotech Conference is one of the most important events in terms of the biotech as it is, and this is a great chance for all of those who are engaged in R&D to get exposed to new knowledge and exchange their experiences and practices. For 30 years, the National Center for Biotech became the most advanced center that has contributed into the development both of healthcare and biotechnology. The background of the center is the history of innovations, technologies that have become the foundation for greater achievements in terms of science, medicine and agriculture. The initiatives made by the National Center of Biotechnologies, which is one of the leaders in applied science and fundamental science in biotechnology in Kazakhstan, has always been supported by the Ministry of Healthcare. The Ministry has approved applied standards and food safety and biotech for healthcare, the developments in cell technology, tissue engineering, and the clinical studies for safety, for instance, were absolutely successful. As instructed by the Ministry of Healthcare, the National Center for Biotech has developed the immune-modifying medication targeted to cancerous cells and treating lymphoma and leukosis, as well as having the fibrinogen gel that contributes to treatment of knee joints in patients who have osteoarthritis. And that medication has been going through the test trial successfully in the National Center for Traumatology. So the center has been reinforcing its international cooperation in June this year through the National Center for Biotech, together with BGI Group, which is the Chinese company, opened the clinical lab aimed at having early diagnostics of inherited and genetic diseases, to include Down syndrome, Edwards syndrome, and other genetic diseases. Previously, while there was a risk detected on having the deviations and to verify the diagnosis, the patients would need to go abroad to have the disease testified, and now we have the opportunities for early diagnostics of diseases in Kazakhstan. Dear colleagues, I hope that the efforts aimed at reinforcing our opportunities in terms of biotech will contribute to the future of our country. And within the two-day event, we will have perspectives in terms of further development and have professional ties established. And I have no doubts that we will open new horizons for the progress of biotech both in Kazakhstan and beyond its boundaries. And I would like to wish you success. Stay tuned and inspired, and let your knowledge and experience contribute to the future of our society. Thank you. Thank you very much, Timur, for such warm words. And now allow me to give the floor to General Director of Fast Biopharm, Mr. Abdrahimov. Good morning, dear colleagues, and good morning, dear scientists. I would like to greet our young, talented scientist first. And today we're going to be celebrating the 30th anniversary of National Center for Biotechnology, which is the most prominent center, and is the foundation of our programs and curricula. So this center actually has become the magnet that has brought together a lot of attention to the center, participants to the conference and their guests, dear friends. On behalf of the National Holding Kasbah family, on behalf of myself, I would like to greet you to the international conference devoted to the 30th anniversary of the National Center for Biotechnology. The new conference setting and undoubtedly is really leading organization in biotech in Kazakhstan. We have witnessed industries, and it gives a great chance to train the new wave of talented experts who will be promoting the science. And I would like to thank the organizers and the partners to the conference for their invaluable contribution and efforts. And your presence here actually is one testimony of the importance of this issue. And I would like us to have fruitful cooperation and let this conference be the starting point for the new endeavors in biotech, healthcare, environmental protection, agriculture, and biosafety. Thank you. Thank you very much, Evgeny Basarovich, and long ago. The floor is given to ex-director of the National Center for Biotech, Mr. Momin Aliyev. So, Mr. Momin Aliyev, please, the floor is yours. Colleagues, it's sad to remain untouched. I would like to greet you all. I have a feeling as if I'm back to my family. We have reunited finally, and each and every one of you actually has witnessed a long way forward. I had 3 major surgeries, and we went to go through hard times as well. I used to be the director of the lab and the director of the National Center for Biotech, and we actually managed to do impossible. And we've been the pioneers in biotech in Kazakhstan, and we have invented the modern language. We made biotech our reality, and I was so pleased to work with you. And the state of Kazakhstan has been providing a great deal of care, and this is a great sign that the government is paying its utmost attention to biotech. And biotech is future. It's absolutely not possible to imagine the future without biotech. And today we're going to be feeling even more happiness. Now, let me tell you that the advisor of the president for science-related issues was supposed to be here with us today, but unfortunately she couldn't make it. And now, let me give the floor to Mr. Abdurrahimov. So there's a technical delay. Zakaria couldn't make it, so on behalf of Ms. Advisor, Mr. Abdurrahimov is going to be making a speech. So, dear colleagues, as it was said by our moderators, yes, Madam Advisor is sending warm greetings to you. There's a meeting at the administration of the president currently being conducted, so she couldn't make it. But anyway, she would like to address all of those who are present here today, and I would like to wish you all the best in relation to the 30th anniversary of the National Center for Maritime. This is a milestone that we also have to acknowledge, and the National Center for Biotechnology is known for its achievements, and it has contributed a lot in terms of the development of science to include agriculture, medical services, healthcare, food safety. National Center for Biotechnology is well known not only in Kazakhstan regionally, but also globally. So I have no doubts that National Center for Biotechnology is going to keep thriving, advising for scientific matters under the President of the Republic of Kazakhstan, Mr. Zakaria. Thank you so much. And I wanted to actually be engaged in reading, and I actually later on have my PhD studies in Moscow. Why is it so? Because back in 1981, Mr. Australian scientist made a prominent statement saying that we were at the age of the genetic changes within the cells, right? And we were so glad to be in the new world of colors of cells, what is going on inside the cell. I was absolutely thrilled. And we studied the regenerative plants, the analysis using our ferments and so on. And our objective was actually to prove that we could have the sequence, and as a result we had new sorts of plants called bacteriobastoma. And I remember my tutor, he said to me, the future is in biotech. You gotta have the Center for Biotech. And in 1986, they established two centers, one in Almaty, chaired by Mr. Akhojan, and the second one was chaired by Mr. Sabedi. And many young scientists came to Moscow, and there was a romantic time, when discovering the world of biotechnology. And when I was back to Kazakhstan, I chaired the lab of biotech. And back then, many people, prominent scientists, I remember Madam Kotelska and back then, young scientists was back of the young prominent scientists who's here with us today. They were making their first steps in science, and it was Stepanogorsk where we opened the lab. And why Stepanogorsk? Stepanogorsk was a city with restricted access. They were producing bioweapons then, so warfare was prohibited to get there. So US senators Nami Lado actually established a program on decontamination and removing militant agents from occupiers from Stepanogorsk. So I was a chair as a vice president for the lab. when we were living in the times of destroyment, back in 90s, for instance, I was demonstrated the PCR analysis. Now everybody knows what PCR is. We have no doubts. We didn't have any gadgets. We couldn't use the technologies. And it's our way that having no things happening. And now it is a great time. It is a great time of opportunities. So we do have a chance to open new areas in science, and I would like to wish the team of scientists and the young scientists especially to keep being creative, because we can't actually progress without having an appetite for science. So create, and create. Thank you so much. Thank you very much, Ayub Rashidovich, for such long words. You're such a great source of knowledge. You were one of the pioneers who have established the center. And as we can see and heard,
в области биотехнологий. Давайте теперь послушаем приветствие от тех, кто эти биотехнологии использует в своей работе. И следующее слово предоставляется директору Национального научного центра травматологии и ортопедии имени академика Батпенова Бекарисову Алжасу Сапаргалиевичу.
Director of the National Center for Traumatology and Orthopedics, Mr. Bekarisov, please. Good morning, dear ladies and gentlemen, dear colleagues. On behalf of the National Center for Traumatology and on behalf of myself, I would like to greet you. So, dear Vyacheslav Borisovich, on behalf of the National Center for Traumatology, named after Batpenov, and on behalf of myself, I would like to full-heartedly congratulate the team of the National Center for Biotechnology with the 30th anniversary since you were established. Since the establishment of the center, we've been partnering in healthcare. We've been implementing such projects as having personalized in Kazakhstan, development of innovative implants to cure musculoskeletal diseases and pathologies in joints. And I would like to mention about our job in terms of development of composite materials and arginine surface that have been proved as very effective in clinical practice. And thanks to our joint efforts, our patients were getting access to high-class treatment. And your center plays an important role in development of biotech in Kazakhstan and beyond its borders, and we're proud that we're part of those achievements. Your dedication to science inspires us to have more initiatives going. Dear colleagues, on such a great day, I would like to wish the team of the National Center for Biotechnology to keep thriving, to keep developing, and I have no doubts that more ambitious projects are ahead, and I would like to wish success and fruitful cooperation to all of you. Thank you. Thank you so much, Mr. Director. Thank you so much, Oljas Sapargalevich, for your warm words, and of course, the last introductory word is given to Mr. Goncharov, Director of Biophysics and Cell Engineering, Minsk, Belarus. Thank you so much. Now we're going to be giving the floor to those who made a very long trip. So, Mr. Goncharov, the floor is yours, please. Yes. Andrei Evgenievich is going to be making his speech once he is with us in the room. And I would like to ask Janar a question. I know that you have been leading a large lab and you've been conducting a series of serious studies, but if you had such a high-class scientist since the very beginning, Rashid, you asked me a very unexpected question, and there's such a person, I think that our center does have high-profile, scientists in each of the department. But I would like to mention about Mr. Ogay, because I used to be his student before, and now I act as a standalone scientist. And now we are launching the practical part of our event, and now the floor goes to Mr. Ogay, Director of the National Center for Biotechnology. So please, the floor is yours, sir. Could I have my presentation on the screen, please? Dear participants to the conference, dear colleagues, dear friends, I'm thrilled to see you in this room, and thank you for coming. Thank you for being with us while we are celebrating 30th anniversary, and thank you for being with us for 2 days, for sharing your experience, for sharing your practice, and together with you we're going to be having the international prominent event. And my presentation is going to be devoted to the history of the center, to whatever we do at the moment currently, and how we envisage ourselves in the future. National Center for Biotechnology is the leading R&D organization with a biological profile in Kazakhstan, with 30 years since its establishment, and we are known as the leaders in fundamental and applied science in biotech, in order to tackle down issues in healthcare, agriculture, and environment and biosafety. And our mission actually is to have R&Ds in biotech together with the other scientific organizations and universities, as well as to engage companies from Kazakhstan and beyond. And our objective is to turn the industry into high income industry. And now let me brief you about our history. As it was said by the previous speakers, the center was established in accordance with the order of the President of the Republic of Kazakhstan in November of 1993, and then later in 2005 the center was renamed to National Center for Biotechnology of the Republic of Kazakhstan, and it was comprised of the following institutes: Republican Collection of Microorganisms, the National Center for Biosafety, and the National Institute of Plants. And in 2015 we moved over to the new campus in Astana, where we are now at the moment.
P5.
And back to 2017 we created the Academic Center. In 2022, National Center of Biotechnology, including the National Center of Kazakhstan. Here you can find the board directors of National Center of Biotechnology. The first director of National Center of Biotechnology came at the beginning of my career, actually the scientist Mr. Vikens, which worked in the Institute of Biomolecular Technology, and then we've been a head of the National Center. The
following up with which is a famous scientist, microbiologist, who spent a long period of time in Steppenwolfsk on a secret base. He's been the head of governmental programs under the non-disclosure agreement. He's been the head starting from the 1995 till 2000. The next one, Mr. Educator. Now he's the head of a huge beer holding in Almaty. Unfortunately, he sold the custody. And currently, he has a business trip abroad and the assets to share the meetings. The next elected him is Miskakov Anil from 2004 till 2005. He shared the whole history actually of the center, and currently he's the employee of Kazakhstan Institution of Agriculture and Plants in Almaty. We had contemporary campus and development of the biotechnological cluster. The next elected being Mr. Nominally Watt, who's done a lot for the development of our center, and it is worth to highlight that we see the reconstruction of our center under the first government. The next one been Miss Zhumudilova Zoria. She's been head of the center from 2020 to 2023. She's been a great enthusiast. Thank you for your active participation. And Mrs. Kakova, she's been head of the center more than a year. Here you can find more information on the history of our center, which started from the building of the campus. From 1993 to 2015, the major building was located on Waltham Street. Here in the picture you can find the small doll building. Actually, now there we have the national collection of microbodies, and in 2006, when we received the funding for building a new campus, we developed infrastructure for building construction. Of course, we've had some delays in construction process, but then we received the gorgeous building. When we built our center from 2006 till 2008, within the center development program, we organized the workshops for the leading specialists in the country. We worked together with institutions and universities. We gathered in one place the most prospective young scientists who were responsible for the country development. At that time, National Center recruited the young specialists. Some of them you can find right now in the hall. Some of them are now are heads of the laboratories in the country. They developed gene engineering, cell analysis, and so on. They have the business trips abroad in South Korea, Japan, Israel. In 2008, back in July, under the order of Ministry of Science and Higher Education, we opened the National Scientific Laboratory. The Mr. Meneliyev been the head of this laboratory. He impacted a lot due to the fact that he motivated the employees. Each week we had the reports. He taught how to write in articles, and in fact he increased the level of knowledge of the employees. Thank you a lot for the new methods of education and teaching. We've got the most developed laboratory. At that time we've received the highest awards, and we've got the best workers right there. Currently the head of this laboratory is Miss Zholdubayeva Yelena. Now she's a professor who been under the governance of the Mr. Meneliyev. Currently we have 15 contemporary laboratories. If you are interested in them, we can have some kind of excursion and discuss their work in details. The work directions are quite different, from the genomes of plants up to genome editing. We have two buildings in Almaty and Stepnogorsk. In Almaty, we're more focused on biological safety problems. Here you can find some information on our employees. We have the great pool of employees. We have the wide range of young specialists. The average age is 36 years.
Исследователей.
We have 256 scientists. We have doctors of science, 15, and 44 candidates of science, and 39 PhDs. So we have lots of young scientists, and we have 29 of them and 90 master degree students. And the center has been investing a lot in terms of training and training available for students who study elsewhere in other educational institutions. And we've been partnering with national universities, not only from Kazakhstan, but also we have partners from Russia, for instance, and we've been publishing articles together. We've been partnering with the Kazakh Technical University, Nazarbayev University, and with the Kazakh National University, since our doctor degree students and master degree students actually have internships there, and vice versa. So we've been arranging workshops, conferences, as the one we're having at the moment. This is the conference devoted to the anniversary since we've been established, and actually some of our prominent scientists became the PhD and doctor degrees, and we have the internships beyond Kazakhstan in the leading foreign centers from all around the world. And you could see Hirsch index. So this is the reference to the national rating agency called NHR for 2024, and our rating is 3.5. We've been placed to position number 13, and from the biotech centers, our center is number 1. So the average index of Hirsch is 3.5% in total. Definitely, the end result for any scientist is either a patent or the article that other scientists from all around the world get exposed to. So you had the statistics of our publications and comparison as of 2018 and 2003, you know, Scopus, for instance, rating, and we've been publishing in average 60 articles per year, you know, and patents have different tendency because it depends on the stage of completion. The center has been awarded repeatedly for being active in terms of publications. So Web of Science, for instance, has acknowledged us as the most cited organization in 2011 and 2015. So we received the award, Scopus Award, Research Center of the Year, and in 2020 we became the leader in terms of global science for publications. And I would like to mention about the awards given by the patent agency. In 2020, our team of scientists became the winners of Shapagat contest. And my colleague worked together with me, Ms. Naurbaeva, and that was a great contribution in terms of regenerative medicine. And she was awarded with this award. And in 2019, we became the winners in terms of patent, and that award was provided by the Eurasian Association of Patents. So, so actually, that was the work in fibroplastic. And our center has been partnering with a lot of higher educational institutions from 20 countries from all around the world. We have 50 MOUs concluded, and we've been partnering with 34 scientific organizations as well as 11 production facilities. And let me mention about our achievements. So those are the developments that have been made by our scientists. It's a T system for VET, for brucellosis, for EFAT system, for the different types of diseases, to include chlamydiasis as well as the anthrax. And during pandemic, our center was engaged in countering this disease, and we've been working in order for us to have individual PCR kits. And that kit was developed actually within a very short period of time, and that was thanks to the prominent work. You know, we've been loaded and overloaded in order to make sure that we comply with the instructions of the government, which was to have as many kits as possible so that we could diagnose the maximum number of patients. And our scientists were awarded with Халық алғысы award, you know, which was acknowledgement of their hard work. And our branch office actually deals with the industrial biotech. So those are the two major products, the supplements. BioLife, this is the similar to Linux, it's probiotics, the bifidobacterium concentrated supplement, and it uses the gelatin as the foundation, so you could actually have a look, and some of the drugstores such as Alphabet was already buying this. And the second supplement is Biomed, and this is purposed in order to make sure that intestines work better. and you can actually have a look at this supplement, and we have a stand at the exhibition. So Juldabai has developed a T system for an individual dose of warfarin and Flavex. That was the project under the grant of the World Bank, and omeprazole dosage calculations. And we also do have the T panels for the early diagnostics of cancerous diseases, endocrine diseases such diabetes and syndrome Alport. And one of the achievements that we have so far is cell-based medication, and it was developed by Ms. Dailabaeva. We worked together, and we created such a cell-based medication, allowing us to recover sooner after the burns or trauma. So this dermaplasty, this medication is called fibro spray, and they use it for wound healing. So we've also created biomaterials to regenerate the joints and complex fractions. It's a biocomposite material that's a biogel, having some cell growth factors, and in combination it actually reinforces the rehabilitation of the bone. And you could see the pictures before and after. The hydrogel was implanted, and actually we tested it for safety, for therapeutic efficiency, and that was done under the umbrella of the Center for Traumatology. And our objective is actually to make sure that this technology is indeed successful from a therapy standpoint. And this technology was developed by our lab of genetic engineering. Mr. Shustov is chairing the lab, and he's been conducting this initiative. And the Ministry of Healthcare has been planning to have the first clinical trials in Kazakhstan with the use of CAR-T technology that are effective in terms of targeting cancerous cells in case if the patient has the blood cancer. And we have the joint project together with the Center of Transfusion and National Center of Oncology. And as soon as we get the grants, we're going to be having those first clinical trials in patients. And you could see other developments that we have had for food industry. We have a couple labs that deal with these studies in particular and produce these products. And that's amylase, for instance, the fibers that we extract from plants, and this actually could be really good in terms of production of acids, milk acids, and that was funded by the Foundation of Science, and actually you could see those on the screen. So here comes the developments for agriculture. We have 3 labs that deal with the plant breeding, and we have developed a spectrum of different products to include different sorts of wheat, as well as biotech helped us to grow new sorts of potatoes with new characteristics. And we also do have the other developments, the achievements of our genetic engineering in plants. They use CRISPR-Cas So the editing genome in plants, and we managed to have the new cotton called Turkestan that is tolerant to phosphoryltrizin, one of the components of hybrid. And actually, we also have lucerne planted with CRISPR-Cas genome editing, and now we have the new sorts of potato that contains pectin. And then the last plant that I would like to mention, you can store potato under the colder temperatures. And this is the last slide that I have to share. I decided to mention about 4 major statements, which is enhancement of quality of studies, expanding of spectrum of studies in biotech, GLP and GMP, as well as ISO 17025 and ISO 13485. So those are the standards that we would like to comply with in order to provide services and to make sure that our products are produced in accordance with those standards. Number 3 is the intention to develop our infrastructure in order to keep up with the diagnostic systems and the different medications. And we also plan to partner with the educational institutions from all around Kazakhstan and the globe. Thank you so much for your attention. Thank you very much, Vyacheslav Borisovich, for such a comprehensive presentation. Now let us end the introduction part, and now let me give the floor to the moderator, Nazarbayev University NLA Lab Director, Dos Sarbasov, and Mr. Ogay. Thank you so much, Vyacheslav Borisovich, and the practical part of the conference is going to be and National Lab Astana, Dos Sarbasov and Vyacheslav Borisovich Agay are going to be acting as moderators of the session. So dear participants to the conference, now there's going to be the plenary session commencing, and the presentation is going to be made by General Director of National Laboratory Astana, Professor Dos Sarbasov. And the topic звучит как подавление крас-мутантных типов рака, трансформация проблем в новые возможности. So you are given 20 minutes for presentation, please. Good morning, dear ladies and gentlemen.
Ahmed, the organizers. It's good to be back, как говорится, we're across the street. Очень всегда приятно приходить.
It's such a pleasure to be back and see our colleagues and collaborators. I just wanted to firstly share a story. I just wanted to make a commentary in regards to the history. You said that in 2005-2006, National Center for Biotechnology was getting more and more funds, and as a fact, I wanted to say that as a catalyst, it was my professor, Rahmet Hadji Irskinbayev. So back then, he was the Vice Minister of Education and Science, and there were really strong mechanisms of influence, actually. And thanks to him, I started dealing with the science, actually. But I wanted to deal with the fish, actually, and but I wanted to be the director, of fish, of fish factory, So let's talk about more serious stuff. It's important actually to mention about the fact that the science has to be not just, something that gives us a chance to get more advanced, but we have to be advanced in terms of health, in terms of technology and everything. And I just wanted to draw attention to the fact that the first prominent science ever, Kanish Sapayev, is on a screen, and he always kept saying that the science could be successful only should it be working in cooperation with the industry. The cooperation between science and industry is something that we need, and this is still true. And we convert the science, and there's lots going on. We do have the law on science, and we get provided a lot of funds, and a lot actually is done in order for science to thrive, right? And so that we are the catalyst of the development of industry in Kazakhstan. And one more thing I want to share, um, we shouldn't be just developing the applied science. You gotta start and being an expert in in some area. You have to understand the process and the mechanism. If you study just the mechanisms and by doing so, you can get the process promoted further. In the applied manner, so to speak. And this is how the things work. You could use your developments in industry or in other areas. So now let me talk to you about the the science as it is. We work on one of the um, important things, like tackling cross mutant cancers. I deal with the fundamental science, and KRAS is a very small oncogene, but it is really strong. Why this is a G protein that it is hard to target actually, and all of the mutations are related to the functional glycine, there's 12 positions, and over there there's a very strong connection with the 3-phosphate. And actually, so this is the G protein on the screen, and it activates MAP tendency and PFT kinase, responsible for proliferation and the growth of cells. And at that age, actually, 10 years ago, and actually the question was how to block this small pocket, So how to produce the inhibitors. 10 years ago, when I was not dealing with it, I was and I was asking myself a question, how the nutrients impact upon the penetration. So the director of Indiana Cancer Center, um, made an article, that actually put us into the dead end. The question was about transgenic model, So pancreatic ductal adenocarcinoma, it takes only 3 months to have that type of cancer to develop. To your left, you could see, the large tumor, and if you're going to be having dicycling, so express a K Russell, and oncogenic, and then the tumor gets disappeared in 3 weeks. But what makes me wonder is that should we provide more months, then the tumor is getting back. So it's important to have a look at the oncogene, for instance. In half of the cases, the KRAS is leaking, promoted Doppler cycle. It starts expressing itself. And to your right, you could see that the activation, right? So it's KRAS-driven tumors, right? So there's a relapse of the tumors. But 50% of tumors, it turns out to be, that they get switched to activate other oncogene, and it's called the switch to YAP1 expression. And therefore, if you target the oncogene, the tumor is going to be finding, other cells, and even the direct targeting is going to be a problem, right? So back then, I'm talking about the KRAS mutants. Many companies started making special mutations. You could see the GTP bindings pockets, and they started using specific inhibitors. And for G12D or G12C, when you make the specific inhibitors on points, they were very effective. There's really good results in animals and even in human beings during the clinical trials for 2 or 3 months, and then the tumors came coming back. And it turns out to be that the mutations were happening in KRAS, so they actually attack the inhibitors. So it's really, really hard to target this type of cancer. And we know that the challenge is that KRAS mutant cancer cells are addicted to glucose. They're very, very hard to be attacked by chemo, for instance. And what we saw is that the workload effect is really, really, great. There's like heated car, if I can use this comparison. So what did we do? We approached it from a very different angle. We wanted to understand what nutrients are needed in order for KRAS mutant cancers to survive. In amino acid deprivation, they survive, without any problem. But if they're deprived glucose, they die in 2 days. And we decided that we need to understand the mechanism of how to kill those cancerous cells, and they can. There's nuclear condensation, for instance, and that mechanism was started to be used as a start off. We proved that there's KRAS inducible. We screened a lot of types of cancerous cells. We made crash leading when we induce KRAS impression, and if the KRAS effect gets expressed, then those cells are very sensitive in terms of glucose deprivation. So we actually proved that, and we started looking at the biochemical things going inside the nucleus. And we divided the fraction into soluble and non-soluble. And what we saw was that actually when there's a glucose deprivation, then HDAC, histone deacetylase complex, and this is a very strong complex related to silencing mechanism. This is something that gets converted into chromatin, and there's a deacetylation of the chromatin, and which is related to nuclear condensation. So that it's histone acylated. So how this is happening thanks to deacetylate. So over here you could see HTK9 and HTK27. K9 is lysine 9 and lysine 27. When they get histone deacetylation, then it is related to the formation of heterochromatins, and we proved that as well. So whatever we see on the screen, actually, while we grow the normal cells, and the next is how we experiencing the acetylation of chromatin, and we also proved that there's a deacetylation of HT9, and the same cycles become become accompanied with the heterochromatisis. And then the most important is that we wanted to understand what mechanism was behind it. We know that in case of glucose deprivation, we have radical cells, And therefore, we want to demonstrate that, what's happening. So they contact with mitochondria, and we could actually block the process with the help of reducing a component called rinacetin acetyl, and we could block the process actually of deacetylation and even the death of cells in case of glucose deprivation. So actually we can lock it, so the entire set of these processes is getting through thanks to the radicals getting developed in those cells. So the model was very simple, in case of glucose deprivation, mitochondria generate radicals that lead to the death of cells, right? And we can not certainly have the glucose deprivation in human beings, because glucose is a very important metabolic agent, but we could use radicals, right? So firstly, of course, we worked with oxynitrioside, it is used actively to target leukemia. And what we saw was that we could simulate the glucose deprivation. Could I have my slides back? Yes. And actually, we simulate glucose deprivation thanks to oxidants. Each duck is recruited by chromatin, and then should we have a larger time for oxide or oxygen trioxide, we could actually have the deacetylation of the stones, whatever we see in glucose deprivation, to your right, we could see what's happening as the nuclear condensation that is happening during glucose deprivation. And we started using oxynitric and looking at how it targets the KRAS mutant cancers, and only there was a high concentration of but that was sublethal concentration, but the KRAS mutant get suppressed. This is what we demonstrated. Conceptually, we demonstrated that oxynitric actually as a monotherapy is impossible to be used, because otherwise you will need to use high concentrations, because this is poison, right? And we didn't see much therapeutic window for that, and we scantily actually published a large article in Science paper, and vitamin C in large doses is the oxidant, and it kills KRAS mutants of cancerous cells. And over here we have the effect of Trojan horse. Vitamin C is antioxidant. So how can it be oxidant, right? So when it gets into the body, vitamin C actually gets acidic, and it's dehydroascorbate, and then this dehydroascorbate, in case if KRAS mutant get absorbed by glucose, those cells, get dehydroascorbate absorbed, and that effect actually is something that gets vitamin C converted back in the cells, into the original formula. So actually, this is happening thanks to cations, and the acid restoration mechanism of the cell works in a way so that the hydroascorbate works, and it impacts upon the elevation of stress. And we decided to use oxynitric together with vitamin C, and together with vitamin C they work really well, in synergy, while targeting KRAS mutant cancers. And actually it works really nicely with oxynitric, and you could see how fast the cells die. And this is thanks to the generation of the large number of radicals, so it's a cross reduction. So we call it a suicidal growth production by mitochondria. And actually we put more wood into the fire, and this is a great effect that we've been studying. So normal cells don't die, and ATO-DVC is actually a great combination. So the model is really simple. We could see how the glucose deprivation works. We simulated thanks to the ATO-DVC, and what's nice is ATO and DVC is ATO-approved component. So now we could breach the clinical trials. And actually, the idea is that vitamin C is the agent absorbing the cations, and arsenic trioxide could act for protection purposes. And and please pay attention to the doses, to dosage 0.3 grams. There was a good process in colorectal cancer, and we had a patient with less growth by 30%. So actually, we're working on the dosage. So we want that patient or another patients to have larger dosage of 0.6. But we need to have preclinical first and prove what is going to be the maximum tolerant dosage of medication. And in that case, we will be able to see the effect. Acknowledge slide. I just wanted to thank our clinicians and Kazakh Center of Oncology, chaired by Adil Kaidarov, and we've been working, very, very actively. We had the program for 2 years, and our ambition is to complete the studies. And I have our flag members, and I haven't mentioned about the generation of in case of ATO-DVC treatment, and we also look at ribosoma. Bayan Sulu, Nargis, and Narujan work in that regard, and we also have Jansaya and Askar on the and Christina and Agata work on how mitochondria actually produce radicals. Thank you so much. Thank you very much for your nice report and breakthrough and breakthrough results. Unfortunately, so if you have any questions, please use coffee break for that. So now, allow me please to give the floor to very well-known prominent Kazakh scientist Sadanov. So he's going to be talking about design and organization of production of domestic original medicines. So he is the general director of microbiology and the Virusology Center. So thank you so much. So good morning, dear ladies and gentlemen. So I would like to congratulate you with the 30th anniversary of your center. So actually this is the first time ever I still remember how we gathered together 30 years ago, when Ayub was our director. And actually we work together with those partners who are here today, and I was the deputy director responsible for scientific development. And actually, we moved over from Stepnogorsk to Astana, and I still remember those days. You know, we used to be working together at the agricultural university within the framework of the lab. And actually, for 30 years, for it's not large amount of time for the history. However, it's really impressive in order for us to have some results, right? So you can actually have a result achieved for 30 years, and we have actually gathered together, and our colleagues used to be, ministers and vice ministers, and they were the pioneers in terms of biotech in Kazakhstan. So in order to have this center built, they made so many efforts. So therefore, I wanted to say that the Center for Biotechnology is not something coming from scratch, and I wanted to again, congratulate you. You have such a wonderful building and wonderful equipment. So biotech was a standalone program. Central funding program, was used for a wide range of objectives. We had to manually actually distribute funds. And we have our colleagues sitting here today, and they know that they're really hard times actually. And again, congratulations. So dear young scientist, all the best to you. And so I shouldn't be making leap talking. National Center for Biotechnology is the business card of the country. There are lots of institutes. The Botanics Institute, for instance. However, the National Center for Biotechnology is a very, well-established facility. And this means a lot, actually. This has to be an institution aimed at providing innovations for the industry, right? They have to produce. They have to produce for agriculture, for agri-industry, for healthcare system, environmental protection, and so on. And thanks to that, the economics of the country has to be growing. Whatever is we all work for. This makes a difference. So from this standpoint, lots of countries were thinking about what to do and how to do. How to provide funds and so on. And what does the state get eventually, right? So this is the major question. What have we contributed, and with what have we contributed with? Of course, it's nice to report, how many labs we have, how many people, how many PhDs, and so on. This is something that we all need, and therefore, I feel happy listening to the presentations, comprehensive ones, really large scale and so on. However, we have to make sure that we do make an accent not only on the fundamental science, but on the applied science as well. We have to shift over so that we could provide new innovations. It shouldn't be just PCR testing, not just diagnostics. That's just the beginning of production. There's a vaccine, there's medications, or biological means for agriculture. So this is the end of the biotechnology. Dear colleagues, I won't take too much of your time. However, microbiology and virology has two schools. So the first one is microbiology school, and virology school. Microbiology school was chaired by a prominent, academician. So, and they have, dynasties of scientists. Shamshir Jamatovich is the prominent scientist of 50s and 60s of the last century. We have to talk about the school. There has to be an experience that would be inherited, inherited within the school. So we have to start with the fundamental. And those Sarbasov made a speech. There has to be a mechanism. So there has to be the mechanism of actions, and only in this case we're going to be having the tangible results. And it's important, to actually be consistent in terms of studies. So in order to have the result, first of all, we have to have scientists, we have to have experts. That's number one. Number two, all we need is the equipment. The next is funding. So we have to provide the result, right? If the state isn't given the result, this is going to be deceiving the state or deceiving yourself, right? So therefore, dear colleagues, we have to be sovereign, we have to be independent. It's not only the country to be independent, but the sciences to be independent. Let me be frank. We've been partnering with our foreign partners, and not a single foreign scientist is going to be sharing the technologies. Never ever. You may purchase the technology, yes, but if you are capable of developing it yourself, do it. And in the end, by the end of the day, the science is going to be stocked should there be no development. So our institute deals with the full cycle to include preclinical, clinical, development, and organization of production. So actually organization of production, especially when we talk about medications, is not an easy thing to do. We have to arrange everything, we have to produce, we have to address the full cycle. Can Kazakhstan actually be in a position of having the medication produced eventually? Currently we have 67 plants, Shymkent, Himfarm, we have 7 facilities in Almaty, pharmaceutical plants. 67 again, I'm reiterating. However, all of those plants use foreign substances. What are foreign substances? Those substances coming is something that is delivered from abroad. This is something that our foreign colleagues develop. They transport them in barrels. All they do is they pack. So packaging is happening in Kazakhstan, and it could be pills, sachet. It could be any other forms of packaging, and then they put a stamp made in Kazakhstan. But it is not the case. The first vice minister left. I would tell him. So currently in Kazakhstan we have only one regional medicine. Only one. The only one in the country. For 33 years, Kazakhstan managed actually to have only one medicine. There is none. Whether there's going to be more or not, the time is going to show us. Therefore, we have a very important task, especially those who are dealing with farm industry. This is a very complex process, and it takes time. Next slide, please. So actually, let's talk about mycosis. You know, the problem of mycos, not only in Kazakhstan but elsewhere. You know, so according to WHO, from 1/5 to 1/3 of the world's population suffers from mycosis. You know, in Kazakhstan, the number of people suffering from mycosis, according to various estimates, varies to 1 million people in Kazakhstan. You know, and in fact, we're getting more and more patients with mycosis, and the patients who come from elsewhere, you know, are not cured. So, you know, mycosis is extensive group of diseases, and they are all caused by pathogenic fungi that parasitize the skin and mucous membranes and other human tissues. Among all skin infections, they occupy the first place in terms of prevalence. You know, and now, you know, we have achievements. You know, we actually made we made the medicine. Now let me talk to you about the registered medicine, which is effective. It is roseofungin ointment 2%. You know, that's antifungal drug for the treatment of skin mycosis. You know, if there's a proliferation through the cell, you know, then there could be the lethal effect, you know. So therefore the question is really serious. The mechanism of action is based on the fact that they interact with the membrane of fungi, causing cell damage, leading to this to cell death. Then the problem of vaginal candidosis, you know, candidosis vaginalis is an infection, a lesion of the mucous membranes of the vulva and vagina caused by fungi. And, you know, Ministry of Healthcare currently doesn't know what to do, you know, and especially the vaginal organs of women, you know. They said that every second woman has candidosis, you know, and now about 75% of women under the age of 25 had at least one episode of vaginal candidosis in practice, you know, and it leads to infertility, you know. And next, it was proved that Canprofam is a medicinal product for the treatment of vaginal candidosis. So now the clinical trials are coming. Are arranged. You know, so this is something that we are giving over to the experts. You know, and the results will be reported later. You know, some of the scientists, you know, share with ideas. You know, it's not you to give the assessment. You know, the human body is to be providing the assessment of the efficiency. If you are able to treat it, if you are able to make sure that from the clinical standpoint 100% of patients are cured, then you can say that yes, this was your success, you know. But you know, the mechanism of efficiency, you know, nobody prohibits you, but you know, and some of the scientists, you know, 99% of scientists start their lab studies, you know, and they finish with lab studies, you know. They even can go through you know, and get to the prominent results. And we shouldn't be just praising ourselves for the sake of praising, you know. And people need the result. The state needs the result. Next. The next problem, acute intestinal infection. So in accordance with the WHO, up to 1.5 billion people are registered annually in the world. So, and 70% of them are children. You know, this is a very serious disease. However, we are now shifting over to the third phase. So the first phase is 100% Second phase is 300 people, and third phase is from 1,000 to 3,000. So if 99% demonstrates positive effect, then there's going to be taken as a result. If not, then at any time, there could be the consequences, So the clinical trials aren't going to be approved. So in our case, the clinical trials are very, very tough. The requirements are really, really strict, and therefore, they sometimes, forget about the initiatives because they don't want to deal with the requirements. So the mechanism of action. The mechanism of action based on probiotic bacteria, in human immune system through the synthesis of B vitamins, essential amino acids, and hydrolytic enzymes. We might not have enough time. The U.S. Colleagues helped us, but we finally proved the mechanism of action, and 2 years later, this medicine is going to be on the market next. ASBA norm, so a broad spectrum drug against human intestinal infections, And so I want to explain that to you, dear colleagues. The essence is in this. Just please have a closer look, especially those dealing with the medications. In order to start and complete the cycle, all the way down to production, the also is to go through these stages. So number one, so isolation will be produce a strain, then drug development, preclinical studies. So it takes at least 1.5 to 2 years. The first stage takes 2 years. The next, is the development of the medicine. It could be up to 6 years. Clinical trials, the first, second, and the third stages of phase. License for pharmaceutical activity. Can you get the license or not? So next is validation. Every single minute, the process is to be controlled and fixed, and there's going to be this special the regulations. If you are able to prove the technology to the health minister of healthcare, only in this case they're able of, giving you the approval. The last one is the organization of the production. So it's 14 stages. Every one takes up to 2 years. So you have to estimate that you're gonna be needing at least 25, 30 years. Those who keep saying that you will spend 2 years, to go all the way through, it's not gonna happen, And in Kazakhstan, we actually converted over to GMP standard. Now we're constructing the pharmaceutical plant in accordance with the GMP standard, and you know how complex it is. The European experts, verify every single detail, every single square centimeter of the facility. So, dear colleagues, I would like to once again congratulate you, and that's great that we're dealing with this science. However, it has to provide us with the results. And in biotech, if there is no result, then it's not possible to name it biotech. Thank you. Thank you very much for your report. And now there's going to be coffee break. So 20 minutes, please. We are behind this schedule. The coffee is served on the 5th and the first floor. Thank you.
После кофе-брейка пленарное заседание продолжится.
конференции, коллеги.
Dear participants to the conference, dear colleagues, please join the plenary session. We're going to be starting off shortly, and the next presentation is going to be given a couple of minutes later. Thank you.
Okay. Our international conference. Our next keynote speaker is Professor Raphael Song from New York University, Abu Dhabi. His presentation, Growing Spheroids in Continuously Perfusible Microfluidic Chips. Okay. Please.
Thank you, Professor Gay. Oh, okay. Thank you, Professor Gay. Thank you for inviting me to this conference. As Professor Gay mentioned, I'm from New York University in Abu Dhabi. This is a new global campus of New York University in the UAE. And I'm very fortunate enough to have a lot of smart students from all over the world. And I have a very, very smart student from Kazakhstan in my group as well. And through the students, I have created contact to Professor Gay. And Professor Gay was kind enough to host some of the students during the summer and enable them summer internship opportunities. So I'm very thankful for his kind offer. So today, I'm going to talk about my research related to microfluidics. So I am bioengineer. I am interested in building devices. I think one of the speakers in the morning mentioned the importance of science. In order to enable science, I'm offering devices, new tools that allow scientists to do interesting scientific studies. And the tools I'm developing in my group are related to microfluidics. Microfluidics is where you create very tiny micro-scale channels to control the flow. So using microchannels, you can create very controlled microenvironment where you can study the onset of disease very, very precisely. And this is a tool that I'm going to talk about today. Before I get started, everybody knows where New York is. The new campus of Abu Dhabi has been operational since 2010. And we have roughly 2,000 undergrad students from all over the world. We have around 60 different nationalities. And the students come and spend one semester in New York and then the rest in Abu Dhabi. So if you have a chance, please visit us in Abu Dhabi. And more recently, in 2013, NIU has also opened third campus in Shanghai. So through these three campuses, we create a truly global network. And students can go from one campus to the other campus and take courses. And in this way, students get really exposed to different cultures, learn different languages, and become really global citizens. And this is the goal of this global network campus. We have also a very beautiful museum in the neighborhood. It's Louvre, Abu Dhabi. And this is designed by a very famous French architect. And this is something that you don't want to miss when you come to Abu Dhabi. So please join and come and visit us and see also this beautiful architecture as well. Now, this is sort of the motivational slide. Often we use 2D cultures to cell cultures to study diseases. So you take a petri dish, and using petri dish, you put cells, and you can study the cells and under different conditions and so on. But the problem with this very commonly used 2D culture is that there is a lack of cellular complexity and network geometry. Because in reality, cells are not lying on 2D surface. They are creating a 3D structure rather than 2D structure. So the ones that we are using in the laboratories are not really replicating the true structure of cells in the human body. If we go to animal models, they're beautiful. They are really replicating the human body. But there is a difference between humans and animals. So even if the animal models can give us a good indication whether this new approach that we are taking could work in the human body, but this animal model cannot really recapitulate the human body. So in order to solve these issues, people have worked on 3D spheroid, on 3D cell cultures, 3D tissues. Using various methods, people have created aggregation of cells and recapitulate 3D structures of human body. One of the most common methods people have used to create such a 3D cell tissue structure is called hanging droplet. It's a very simple method. You use droplet and put some cells into the droplet, and based on the gravity, cells will aggregate at the bottom of the droplet. Now this method allows to create many spheroids at the same time. Now the problem is with this, even though you can create 3D structures, these spheroids don't experience any dynamic perfusion. These spheroids are under static perfusion. And this is not a true condition of human body. In a human body, cells will never be under static perfusion, but rather under constant dynamic perfusion of blood and of any fluid. So even though this hanging droplet method allows to create 3D cell cultures, the problem is that these spheroids are under static perfusion, and therefore there may be some differences between the static and the dynamic perfusion. So what we have come up with is try to create a device that allows us to create 3D tissues, 3D cell tissues, similar to the hanging droplet method, but under dynamic perfusion, where we can perfuse medium through the device and apply shear force to the cell spheroids. As is shown on the next slide. So we simply built a device that has many wells, looks very similar to multi-well plate that people use in laboratory very frequently. So this well plate is open, so you can simply put a certain number of cells in each well. Now the novelty of our approach lies that you can close this device. As shown here, once you load the cells in each of these wells, you can use a adhesive tape with a cover and close these wells. Once these wells are closed, you can simply connect a pressure pump or a syringe pump to one of the inlets and start perfusing medium through the wells. So through this approach, we call this hybrid approach, you can take advantage of open structure of wells with the dynamic perfusion of microfluidic device. So we can solve this problem of hanging droplets using this novel device design. And once we have perfused medium through the wells, and we have grown the spheroid to a certain size, we can simply open the device again and retrieve spheroid out of the wells. So in this way, you have sort of emulated the dynamic perfusion of human body inside a chip. Once we are done, we can simply open and then recover the spheroids or organoids and then study them with various methods. So let me show you some of the designs that we have come up with. So in this case, we have 9 wells. Now using this reconfigurable channel design, we can either connect all the wells in series, shown here in the first. And so we start perfusing the medium through this inlet, and this medium goes through all the wells and then enters or exits at this outlet. We can also use this method to reconfigure the channel design. So instead of connecting all the wells like in series, we can connect only 3 in series. So this is what we call channel configuration tool. And connect only 3 wells in series. Now, using this method, we can also create a different configuration where each well is connected only to itself. So there's no connection between wells. So this is sort of the condition you have in conventional micro well plate, where you have each individual well independent from other wells. We can emulate this also using this design. Now, the beauty of our approach lies that you can reconfigure the channel design very quickly, depending on your need, whether you want to study the connection between cells, or you don't want to study any connection, but you want to see only individual spheroids in each well individually. Depending on the need of scientific question, you can use any of these designs, and this channel design can be quickly reconfigured according to the need. Let me show you a couple of examples that we have obtained using this reconfigurable microfluidic chip. So in the first slide, we use very simple mouse embryonic fibroblast cells and connected them all in series. So this channel configuration number 1 is where you connected all the wells with each other. And this is the result we obtained. In the device, in the microfluidic device, we started out with this small spheroid on the order of 300 micrometers. And after day 14, we obtained beautiful, larger spheroid grown to around 600, 700 micrometers. So we managed to maintain such a large-scale spheroid inside our device over 14 days. And on the bottom, you see this is a spheroid obtained from the conventional microwave plate as a control experiment. You see, in terms of sphericity and in terms of the size, there are hardly no differences, which means our device performs as well as conventional microwave plate. We also tested the breast cancer cell line, MDA, MB231, and you can also see that the spheroid in the device grows to a similar size as a spheroid in a normal well plate. Lastly, we also tested human-induced pluripotent stem cells. And as you can see, in terms of size between the device and, in terms of device and control, you can see the size of this human-induced pluripotent stem cells inside our microfluidic device grew larger than, this is the configuration number 1. The size is similar to the control, but if you look at the morphology of our human-induced pluripotent stem cells, you can see the shape is more irregular because, as you remember, our device allows a dynamic perfusion, which means the device applies constant shear stress on the spheroid. And because there is a constant shear stress, there is a differentiation happening inside the human-induced pluripotent stem cells. And hence, you see now this spheroid out of induced pluripotent stem cells start differentiating on the dynamic perfusion. Whereas, if you look at this spheroid that is grown in a normal control, in a well plate, on the static perfusion, there is hardly any differentiation happening. So which means we can really see big difference between dynamic and static perfusion. And this is, I think, really exciting result that we have obtained using our device. Let me go to the channel configuration number 2, where we connected only 3 wells with each other. And here we can also see that the size of the organ or spheroid is now larger than the spheroid grown in a control plate. And because of the dynamic perfusion that I mentioned earlier, you can also see some irregular shape happening, which means there is a differentiation taking place because of the shear stress. In the channel configuration number 3, where we had only, where there's no connection between wells, we noticed that in the case of this breast cancer cell lines, MDA MB231, the size of the cancer spheroid is smaller than the size of the cancer spheroid in a normal micro-well plate. And this was quite surprising because we expected, because of this dynamic perfusion, we would get larger size of spheroid, but it turns out the size of the spheroid under static perfusion is even larger than the dynamic perfusion. And this is something that we have not really fully understood, and then this is something that we're going to explore in the future. Okay. We have measured the size, but more interestingly, we were quite interested in finding how the internal structure looks like. So you create this 3D structure out of cells, and we were curious to know how the cells were aggregated inside this 3D structure. So the method we apply to analyze the internal structure is called optical coherence tomography. It's a non-invasive imaging method that you can use to look into the tissue up to a few millimeters. And you can look internal structure of such a 3D structure. And we use this OCT to look at these internal structures. And this is the image shown from MEF. And you can see how densely the cells are packed inside the spheroid, which indicate the spheroid is really strongly attached to each other and aggregate to each other. We did a similar thing with human-induced pluripotent spheroids and the breast cancer cell lines. And I want to draw your attention to this one. This is a spheroid grown from human-induced pluripotent stem cells. Now the size of this spheroid is much larger after day 14, and you can see these dark spots inside this spheroid, which indicate that the cells inside this large spheroid are becoming slowly die slowly, which means because of this large size of spheroid, there is not enough flux of nutrients from outside to the inside, and therefore cells in the middle are slowly dying, and that's something we can see using the OCT imaging. Whereas if the size of the organoid or spheroid is small enough, the cells in the middle are still surviving because of the small size. So this is something that we have to solve as well. So vascularization of such a 3D structure is really a hot topic in the community, and people are thinking about how to create vascularization inside this 3D structure so that we can make this organ or spheroid even bigger. Currently, we are trying to create also brain organoid. Now, this is something that we are quite excited about. So we are trying to build a human brain-like structure. It won't be the human brain, but it will be like a small scale up to 4 or 5 millimeter size. And this could mimic the human brain. And using such an in vitro model, we can study various neurological diseases in vitro. And this is something that we are quite excited about at the moment. And this is something that we are conducting at the moment in my lab. And we can see all these different neural markers inside. And so indeed, this 3D brain organoid could recapitulate some of the aspects of the human brain, not in the full capacity, but some of the aspects. And using this, we can do various studies to understand more about the onset of diseases and the causes of these diseases. So let me summarize my talk. So we developed a hybrid microfluidic device that can be opened and closed. And by using such a hybrid concept, we can take advantage of open design plus the continuous perfusion in the microfluidic device. So we can take advantage of both worlds inside the same device. And we notice, because of this constant perfusion, the size of the spheroid becomes larger compared to the static perfusion. But in some cases, such as the breast cancer cell lines, we notice the tendency is just the opposite. So instead of expecting bigger size, we saw a smaller size on the dynamic perfusion. And this is something that is very intriguing, and we are trying to understand why this happens. And this is such an ongoing project in my lab. Okay, I would like to thank the sponsor of my project, and I would like to thank Pauline, who is a bioimaging specialist, and she helped us with the OCT imaging. Thank you so much for your attention.
Dear Professor Song, thank you for your nice presentation. So, anybody has any question to the Professor Song?
First of all, thank you very much for your lecture. I would like to ask whether you do co-culture for your spheroid cells.
Yeah, this is something that I think it's something ongoing. So we're trying to vascularize such a spheroid by adding a second spheroid from different cell type, such as endothelial cells. So we're trying to co-culture a normal spheroid plus second spheroid of endothelial cells, endothelial cells, and try to merge them in the same well. So hopefully, maybe the cells, endothelial cells, will migrate into the regular spheroid and start vascularizing. And this is something that I think could, help us to, make this spheroid even bigger through this co-culture that you just mentioned.
Okay, thank you very much. I have lots of questions, but the most important one, can I ask whether which kind of polymer you use for the cover? Because the oxygen that is dissolved, that comes with the constant perfusion through the media, is not enough probably. So you have to have a polymer that makes enough diffusion of the oxygen from the environment.
Right. Yeah, that's a very good point. We use PDMS, which is a porous material, and it allows the diffusion of oxygen.
Okay, yeah, we used it too. Thank you very much. Thank you very much for your lecture. It was exciting.
Thank you.
Okay, thank you very much, Professor Song. Дорогие коллеги,
So, dearest colleagues, the next reporter is Mamanaliyev Bulat Timirgalevich, who is going to be talking to us how to convert the idea into the medication. So, please, sir, the floor is yours. Once again, good morning, dear colleagues. I just wanted to mention that my unplanned report was very emotional, but at the same time, those who is not remembering the past has no chances for the future. So I'm so glad to have Mr. Manaliyev, Bisimbayev, Iskakov with us here today. You know, we do realize that we are covered by your care, by your warmth, and we can actually address you at any given time. So I think that it is worth mentioning about Rustem Omarov, our distinguished colleague. And we're missing him, you know, it's sad that he is not here with us. So actually, in any way, he has continued his work in his students. And yes, he's not with us physically, but he's with us spiritually. And now let's go back to my presentation. You know, you could ask a question, why biotech, you know? What is the connection? You know, I needed to make a report, you know, and I can comment about what I'm dealing with. You know, I'm dealing with the regulations, you know, so actually this is the FDA of in US, and I realize that there's a critical mass of not being able to understand what the medications are all about, and this is the something extra. We have the scientist, who has an ideal product, He made a nice test, he identified, everything, but he doesn't understand why doctors can't just take it for treatment, Everything is fine, it's been proved and assured and so on. So from one standpoint, this is the case. But we try to explain, in order for the doctor to use the product, the product is to be in compliance, with all of the requirements, the tests have to be paid for. If the product is not included into the clinical recommendations, then sorry, this is going to be done at the expenses of the patients. So there's a procedure called registration of the medication or the product, and after that, it could be actually offered to the market. And this is something that I've been dealing with. For me, biotechnology and the medical biotechnology used to be the story about money. Should this be a story of success, it has to bring money, right, to those who has developed it, or to those who have managed to invest, a certain amount of money into the product. I checked the websites. There's a Skolkovo Park, tech park in Russia, and for instance, there was a test. One of the participants to the conference is directly connected to the company, Mr. Maxim Filipenko. For instance, you could have a look. The amount of investment was immense. That market actually. I made my estimations back in 2023. There were procurement in the amount of 1 billion rubles. The investments are pretty interesting, and this is something that started from a certain moment of time. Those are the people whom we know perfectly well. So they have the kit of reagents, and it's all about money. Money that is paid back, and you can get your investments back. So that company was established by a scientist from Buryatia, and that company actually made over 1 billion ruble. They were selling IFA, to identify the antibodies of coronavirus, and now they've been looking on having that test used for HIV/AIDS. Identified. This is the open info. You could get acquainted with the success stories of the companies, and overall the turnovers are the true reality. Your objective is to pack your product into the package that everybody needs, and the procedure of procurement in Russia is open and transparent. But there's another option. How can we use the Kazakhstani developments in order to penetrate Russian market? Because Russian market is pretty large. We have to understand the specifics of the terminology. There's medical devices, medical products. So medical products is ultrasound, CT, and in vitro tests, kits, and substances working with the samples. Allowing us to provide a diagnosis or have the screening conducted. That's something that is penetrated with lots of terms. You don't have to be scared of those terms. However, should you enter the market, you have to understand. You have to understand what the sensitivity is, what is the limit of identification, and so on. So when we actually set it up, the requirements that would need to comply with international standards, it turned out to be that the specificity is something that matters. For instance, what they would do, they would just mix it and say that the specificity is something that has to do with the likelihood. In 95% of cases, it has to be the case, the indicator. And we have to make sure that there is a right design from scratch, from the very beginning, so that nobody loses, chances. And actually, what is the ways to legalize medical devices? And I don't know if you have the lab tests, for instance, at least in US, this is something that exists. We have medical devices for in vitro diagnoses, and there could be the potential, potential to utilize for the lab tests, for instance, right? And this is the simplest way forward, and we have to think whether there's such an option in Kazakhstan. If it doesn't exist, then perhaps we could have a look at perhaps having an option or having the scheme so that it is supported with the legal framework in terms of the lab tests, and by doing so we could actually get the absolutely legal device, and the doctors could use your device, and could offer it to the patient. So the lab tests, especially those that were developed by Charité Clinic in Berlin, this is the vast market, and from in the United States, for instance, the lab tests kept developed, and the skin that they would use was demonstrated is demonstrated on a screen, clear, would provide testimonies that everything was in compliance. And in 2015, FDA reviewed that there's some tests that comprise a threat. For instance, when there's a neoplasty diagnosis, lab test purpose, there was errors found, and since then FDA thought of maybe there has to be a different approach, and FDA has to be somehow engaged, somehow involved into the process. And in Russia, from the very beginning, they decided in order for the lab tests that have to do with non-registered medical devices. So, however, the document has been simplified, as they can't have, orphan disease patients, for instance, patients with the orphan diseases. It's not possible to have a population for the study. It's 1/10 people. And there's a way to legalize, right? And there's certain requirements. I don't know whether it is applicable for Kazakhstan or not, but anyway, I'm commenting on whatever is happening in Russia. So let me touch upon the new opportunities. We have the new market of CIS. The committee has been working for a long time, and it has been generated a lot of documents. So in theory, should you register your medical device in Kazakhstan, and in accordance with this procedure, you could be acknowledged. I mean, your device could be acknowledged and legalized in Russia. So, and you could do whatever possible. You could participate in open tenders, in open bidding process. It's important for the medical device to be registered in one of the CIS countries. So, I wanted you to pay your attention to this implementation process. Why? Because the market of Kazakhstan is somehow limited. You got to be expanding your presence. And since this union exists, Eurasian Economic Union, you have to just use the mechanism, and then thanks to that, you will get the acknowledgement in Russia as well. So the procedure is not that complex, and the website of the Eurasian Economic Union does have the requirements, and the list of requirements, and what needs to be done, what is recommended, and that's a actually roadmap that will navigate you through this legalization process. In terms of the life cycle, developers are responsible only for the starting point, but the life cycle is much longer, all the way down to the utilization of the medical device, and we deal only with the samples of the patients, so therefore these requirements do exist. And other issue that I wanted to touch upon, and why did I call you to use it? You know, there's standards. You know, ISO has developed different standards for medical devices. You know, as well as they do for biotech. You know, if you are following the standards, then your medical device is going to be safe, efficient, and recommended to the patient. So therefore, you know, compliance to standards is important. So I made reference to certain standards, and this is something that you could use. They have to do with multiplex PCR, digital PCR, and digital PCR, and number of PCR is the most developed instrument. And whatever is inside, this is something that you could assess. There is requirements in terms of efficiency, what characteristics you need to include into the medical device, properties and so on. If you have the plans to get registered, then you need in any case to comply with the standards. You know, this is something that will help you to, number one, have a high quality medical device, and there's a good chance of being registered. And registration, you know, talking briefly, is the preparation of dossier and getting prepared for clinical and clinical, you know, and Eurasian Union is just clinical, so it's a bit more simple. So in terms of the standards, if you are interested, I'm ready to share. If you need those, please do so. We speak about the legal documents. All of the countries have agreed about having, you know, the characteristics, the sensitivity, you know, the limit of and the chances of getting harm to the patient is something that is to be encountered. So, you know, we may, what this means is that if you get the one sample, the results are going to be unified. And to close out, you know, this is a well-known picture by Michelangelo, the creation of Adam. So, actually, people have been looking for different logical, you know, lines, and somebody said that the Lord is the brain, you know, and the dura, right? And we are concerned why they're touching each other, you know, the finger of Adam and the finger of the Lord, you know. You know, they say that the sacral can have nothing to do with the human being, you know, but you could see the developer and the medical industry, you know, the clinicians. So why are they touching each other? Is that, you know, it's all about registration of the medical devices. So, you know, the point of touch is the registration, you know, so there shouldn't be any obstacles between those two. So I would like to wish you, thinking about the registration, let us operate and use the same terms so that you can develop the device in the right way. You know, I said that we still keep being the members of the same family. So please, let's reunion and let's get help to each other. Thank you so much and all the best. Thank you so much for such an interesting presentation. Do you have any questions? Okay, use the mic for that. Thank you. Thank you so much for your report. My question, for instance, in Russian, do you have LDT registered labs or not? Yes, we do. And we have a quite large number of national institutes, like Institute of Hematology, Institute of Oncology. Of course, they do have the potential in terms of studies, but you know, in order for them to be free of the investigation committee, you know, we decided so that they don't get registered in addition, you know, so it is subject to standards of medical services, so it has to be reflected in the medical recommendations, you know, and actually we speak about non-registered medical devices, for instance, and in order to legalize, you know, those scientific centers, followed the steps of getting the registration for non-medical devices. 2 or 3 permits were provided, but we hope that this year the number is going to be increased this year, hopefully. Thank you so much for your question. And the next presenter is a prominent, well-known Kazakh scientist, Yerlan Turizbekov. He represents Institute of Plant Biology and Biotechnologies. So good afternoon, dear colleagues. So first of all, I would like to congratulate you with the 30th anniversary of the National Center for Biotechnology. Let me congratulate you with this date, Vyacheslav Borisovich, Professor Guy, and the entire set of your committee. Thank you so much for inviting me and for putting this conference together and organizing it at such a high level. So in your introduction, you mentioned about the relationship between the National Center for Biotechnology and our Institute of Plant Biology and Biotechnology. We used to be a subsidiary company before, and since then, those times, we keep the really close ties, and I'm honored for you to participate in our 30th anniversary celebrations that we have arranged just recently. So thank you for being with us and following up on whatever we do, and we're proud to be your partners. And we try to participate to your conferences as they are large scale and really important and considerable. Our general director couldn't make it for this conference, but our team members, wrote down really warm words to you. So please accept this book of warm words to you. So today I would like to spend a bit of time so that I could brief you about what we do in our lab. Together with the colleagues from all around the world. Okay, so we have lots of cultures that we deal with, but the major one is wheat. And you see the breakdown in terms of areas and the productivity where we grow wheat. And to your right, you just see the map. And the largest area in terms of wheat production is in the north of the country. And it is up to 85% of overall areas that we use for wheat production. And in south and southeast, we do have the areas where we're capable of having full year cycled wheats, and we have great achievements in terms of assortment of wheat. And at the same time, the productivity still remains pretty low. We have registered mutations year to year, to be honest with you. And this picture depicts what is happening worldwide in terms of wheat grain yield. And the producers actually have been facing a lot of challenges, despite the fact that they had great area. Large areas, and Kazakhstan and Australia are standalone, actually. They do have lots of opportunities to have winter sorts of grain, and to include wheat. And we have the best quality of wheat. And why do they import? Why do they import so largely? Is that the productivity, despite the fact that it doesn't leave much to be desired, the quality still leaves much to be desired. Compared to the other neighboring countries, our wheat is the best. So actually, looking at the spring wheat and the diagram, you could see that the productivity is twice as much as in neighboring countries. And this is related to the technologies and the opportunities that we do have in a country. compared to those opportunities our Australian colleagues have been exposed to. So the last column is Kazakhstan. So this is potential wheat yield worldwide. So the potential in Kazakhstan is to have up to 3 tons per hectare. So in the future, actually, we do have pretty nice perspectives. We can produce more wheat in country, and therefore, for this purpose, we need to do whatever possible in order to strive for better, agronomics, other tools. This is needed in order to make sure that our breed efforts are successful. And the last one is to accelerate breeding program based on new technologies. Our objective is to have those sorts of wheat that are capable of getting through the obstacles, to include those of the environmental nature. So we scientists have to do and take the efforts in order to make sure that the instruments are available. So I have listed some modern genetic technologies for mining genes associated with wheat yield improvement, and I wanted to pay attention to identification of efficient S&P markers and market traits associations of agronomic traits using genome-wide association study. And over here you could see genome-wide association study, and there's a great massive of genotyping and phenotyping, and certainly we do have lots of statistics-based program in order to make sure that there's a direct association between the genomes. And in our papers we use both spring and winter wheat, and just recently. And we have, grouped and made the mapping study. We have a standalone report made by Akirke. So you could see the reference, so it's nested association mapping. So let me talk to you more about the papers that we have both on spring and winter wheat. There's information in regards to the populations and the wheat collection studies. So actually here comes the information in regards to collaboration with the representatives from all around the world. And on spring wheat we had worked within the framework with the EU-led project, and we created a team consisted of 20 organizations representatives called Adaptovit, and our lab also participated together with our breed experts. And the second project is winter wheat, and there was international workshop. We engaged the UK, Central Asian countries representatives, and we worked on having the collection of winter wheat for Central Asia. It's not just Kazakhstan, it's Kyrgyzstan, Uzbekistan, Tajikistan. So we collected a great population actually for Central Asia actually, and breed collection I mean, so that we could pay our utmost attention to winter wheat. And we've been actively working in breeding with the help of Mr. Begunov, who used to be working for years in a row with FAO, with other international organizations, in order to make sure that there's a new line of winter wheat that could be good for south and southeast, and maybe for the northern part of the country. And over here you could see information in regards to the collaboration of breeding breeding organizations, and Karabakskaya schools, and the institute named after Barayev, located in Shortandy nearby Astana, Karaganda, Research Institute, and Institute of Agricultural Studies, as well as Rice Production Institute named after Zhakaev, located in Kızılorda. Despite the fact that those are specific institutes dealing with rice production, for instance, we do have experts who've been looking at the results of our papers in wheat production and actually and wheat trials. And we've been partnering with our colleagues from Kyrgyzstan and Turkestan region. And of course, we made a phenotypic variation of wheat collections in every studied environments in Kazakhstan, so that we coordinate the actions and have unified methodologies in regards to what to study, how to study, and when to study. And we also had the analysis, so Manhattan and QQ plots for days to heading, and we studied the associations that are important for the characteristics. So the time of flourishing is identified on the screen, and one of the associations have been pre-identified, and we should be remembering that there is a great deal of different markers. So actually you could see the chromosome of wheat and S&P markers were genotyped, and associations were studied accordingly. And here comes the general picture. I don't want to be commenting on every single point of growth, but overall I wanted to say that we identified 149 markers, market trade associations, and we keep working on them at the moment. And there were a lot of problems identified. To verify the results is one. We have to understand what associations are totally new, so that while having a validation and verification of the results, we could use them later on in our studies to include different types of mapping and so on. So over here you could see the fundamental studies, and in terms of it applied science, we transformed the significant S&P and cusp errors, and those are the specific markers, and we use PCR imaging and proved that those markers were more effective and more considerable, for the breeding process, for those who deal with the breed traits. Why? Because we could actually impact upon the cell, with the spectrometrum, there's dyeing agents, and we would use electrophoresis, for instance, as to have a look at what's going on to the grain traits. And the isolations, different conditions get imitated, and area A, for instance, could be positively associated with the productivity, for instance. Then we could, make an assumption that there is a correlation to the yield. So we now have created a chain of markers, in terms of sustainability, tolerance to diseases. Out of this is the generated CASP errors based on GWAS studies. So we use it to get passport papers, and we've been working actively with the Ministry of Agriculture, and we've been phenotyping the list of traits, and we provide services for our colleagues from Russia, and some organizations as Omsk Agrarian University is our partners, and in the future our intention is to have it used for practical purposes, for breeding. And just lately, last month, we launched a program on using GWAS technologies in other programs. It's not just the wheat, but other cultures as well. We've been working on these initiatives, and we have lots of objectives, and 15 organizations from all around Kazakhstan have been participating to this program, and Almagul is here. Professor Kakimzhanova is here with us. She has been participating to the program, and we also do have lots of breeding organizations. There's a team on speed breeding, and we could use actually the capacities of other organizations who can generate 3 to 4 generations of wheat through a year using different methodologies. And you could see those whom we've been collaborating with throughout Kazakhstan. Those are just wheat breeding community in Kazakhstan invited to participate in this project. So we have different cultures, like corn, soy, and so on, in addition to wheat, And within the framework of this work, we would like to invite our colleagues so that we could discuss different technologies and answer the question how to use those markers effectively. So Simon Griffiths is our key foreign collaborator for GWAS of breed wheat, and Mr. Margunov. Although he is the resident of Kazakhstan, he is the citizen of the United States, and he's been helping us largely to coordinate our breed studies from Kazakhstan. And we have a couple of breed experts from Skolkovo there, the citizens of France, but they've been very actively working in terms of our studies, breeding studies, as we need lots of tools on bioinformatics in order to use correct algorithms so that we could get concentrated on those technologies and chains of supply and so on. So actually the system has been described, and you could see the integrated genomic selection approach, to include speed breeding. So this is our colleague from India, Dr. Bashnia. He works in Australia at the moment. So in his article on one of the cultures, he put down that should there be a good organization, the genome could give you up to 20% of success and productivity, in different grains. We will use his approach. And to conclude, I'm not going to read it through. Actually, what I would like to say is that it is a high time for us to actively use new technologies in breeding, as our colleagues in Western countries have been using them with a high deal of effect. And now I would like to thank the Ministry of Science and Higher Education of Kazakhstan for the programs that they've been provided grants for. And actually getting funding is really important, and we were exposed to such a great deal of specific equipment, so we're very thankful to you, our dear leaders. And I would like to thank our breed experts, and thanks a lot to our labs, to molecular genetics lab. Thank you so much. Thank you for your attention. Thank you. Yerlan Kinizbekovich has made a very comprehensive presentation. Yes, indeed, production of grain is strategic for Kazakhstan. Yeah, there's going to be questions. Yes, just a couple of questions. We are behind the schedule. Danilenko, General Genetics Institute under Vavilov. Named after Vavilov, Moscow. So you talked about the tactics of using genome technologies in breeding, and you told us about the existence of other technologies. But what do you think about non-genetic breeding? It could be, used to adapt. So microbiomes of soils could be used as well, that are in contact with wheat or any other plant. So in some areas I heard that this is something that you use, but in your opinion, is it a perspective? Thank you so much for your question. Well, epigenetic studies is something that we haven't been dealing with. I know that in Russia there was a symposium devoted to epigenetic genome science, right? And just recently you had a large-scale symposium. Yes, indeed, this is a very important area. However, it has to do with technology. We have to use some other technologies. I think that slowly but surely we're going to be tackling this down. But our objective is to find common understanding with the breeding experts, because you could actually have a negative impact upon some characteristics. We have to understand what the preference, the priority in terms of the technology is. It's important for the breeding experts as the major operators are aware about, accepting and all additional technologies will be for the good. And one more question. I have a technology question. And those markers, were this is some S&P that cover haplogroups, or somehow, distributed through the genome, and taken into account the preploidness of wheat. Wasn't it that something that would have an impact in terms of statistics? Well, thank you so much for your question. That's a wonderful question, actually. Well, to start with, I have to mention about how much has been done at the international level, right? And this is the international project. This is the consortium that was comprised of 30 plus countries, right? And we had to find the polymorphism on each and every image. We had to actually convert it into the into the database and so on. And on, we worked with the European, and this is something that we are very proud of. And actually, the question is the right question to ask. Nothing is done just out of scratch, and we've been using the tools that have been developed before us, and developed by that international consortium. And could I ask you a question, actually? Yerlan Knisbekevich, so we would like to start off moving to genome sequencing of wheat, to have a full genome sequencing. Are we getting a telephone call or what? So is it a lunch break? No. So the question is, so the full sequencing, we have sequence analyzers, and we get the solvents for that, and we have Dr. Kizhanova is ready. And tomorrow I plan to go to Barayev Center. And my question is that, the sequencing, the genomic sequencing, is it going to be given any impetus in terms of breeding? Is it going to be something that is similar to the animal breeding, the breakthrough in breeding? Well, of course, every single population has its own characteristics and special unique features. For instance, our Kazakh wheat has its own niche in terms of eco-geographic characteristics, and for that niche, the breeding experts, made their choices. And the first stage of the studies is the training population, trait population. We have to have a look at the maximum spectrum of sorts.
Сложности максимально.
So it depends on the opportunity to have the maximum polymorphism for the search and this association. Segregation of our catastrophe ones could give us a certain result, but it depends on the segregation level, because we need to take into account any polymorphisms that could be found out, could be connected with those who we identified before. That is, there is a need to avoid any duplication. That is why we created an effective set. So it depends. Of course, I hope that we will have the planned fruitful discussion further. Thanks. I would like to continue our plenary session. We'd like to greet the Dr. Josef Shoki, the Institute of Medical Microbiology, Szeged University of Hungary. The topic of the speech is recent developments in the examination of biology and antibiotic resistance mechanisms of anaerobic pathogen.
First, on this irregular slide, I want to ask your invitation and draw parallels between Kazakhstan and Hungary. So both countries acknowledge the importance of biotechnology early, and once we were rich steppe people. And I will talk about bacteroides. So I know that a group of here are also working on anaerobic bacteria, and maybe my talk will be just a little more basic than the previous ones. So the partition of my talk is just to mention the significance of bacteroides in health and disease, and I will talk about my research topic, the antibiotic resistance mechanism of bacteroides. So why they are important? They are opportunistic anaerobic pathogens causing very important anaerobic infections. They are part of the normal flora in high abundance, and one species accounts for less of the bacteroides in the gut and more in the infections. And they are type organism of the anaerobic bacteria, and they belong to an ancient bacteria phylum. So what is their importance in health? They are commensals, they produce carbohydrate utilization enzymes, and they are responsible for pathogen exclusion. They are endosymbionts and just contribute to the maturation of the gut by interfering with enzymes to utilize Foucault's chains from the newborn babies when they are just switching from breastfeed to solid feed. And by these bacteroides are established lifelong there. And they are immunomodulators by the capsular polysaccharides. And the virulence factors of them are the capsular polysaccharides, so it is a little controversial, but I will talk about this. And some other factors, and I just listed them, and I would turn to the capsular polysaccharide issue. So we know, and I showed that the abdominal abscesses are caused by bacteroides, mainly Bacterius fragilis, and it is mediated by this capsular polysaccharide, which can be large, small, and electron dense. It is shown on the right. And if we isolate this material, it can also cause abdominal abscesses, and it is mediated by T-cell response. And these polysaccharides have a special structure, Zwitterian structure, a positive and a negative side chain, and by these they are capable of making the abdominal abscesses. Having these chemical structure for these polysaccharides, they cloned the genes, and later it was when we had the genomic sequences, it turned out that there are some more other operands for these to produce, and they also observed that they are can be switched on and off by invertible promoter regions. It is depicted here, and sometimes so there could be produced in one strain with higher mobility switches and lower mobility switches. It is depicted on the right. And by this switch on and off mechanism, they can escape the immune system because it's a molecular mimicry, and they can be as 256 variations by this process. And other bacterides can do this also, but the oral bacterides cannot, so it is specific to gut strains. And the other side about pathogenicity, these capsular polysaccharides contribute to the normal development of the immune system. I just listed some issues of it, and we know that by these they are they are anti-inflammatory, but metagenomic studies implicate that there can be causing inflammation also, so this issue is not fully investigated. So my topic, the antibiotic resistance of Bacteroides mechanism. So when we talk about this, we mean the effector genes, the activation, the genetic expression of the effector genes, and the genetic elements, they are mobilized, and the expression regulatory genes. So they can be on conjugative or mobilizable transposons, which differ in the conjugative activity. And the main gene regulatory elements are the so-called IS elements that can be found on the chromosome, and sometimes just skip on the upstream region of the antibiotic resistance genes, and an outward promoter can activate the antibiotic resistant genes and then drive a high-level expression. I just put a slide about the antibiotic resistance prevalences among Bacteroides. There are some very high incidence, there are some moderate incidence, and there are very usable antibiotics for Bacteroides. This is the first slide on the beta-lactamase resistance. So Bacteroides usually have the so-called molecular class 2A antibiotic resistant genes, and many species can contain them. There are several genes for it, and the SAPA of Bacteroides fragilis is the prototype for them. Some are on plasmids, but if we take into consideration in this left bottom pictures the expression of the antibiotic resistant gene and the resistance level are not fully correlated, so there must be other factors behind the resistance. This slide just explains this. So we have the second gene, the CFXA gene, which codes for sulfoxetine resistance, and it is on a mobilizable transposon. And if we examine the resistance level and the gene positivity, it is a overlap between the CFXA negative and positive strains. This means that the CFXA gene is not capable to account for all the resistance patterns. And on the right bottom, it is a so-called competition assay, which just roughly means that with increasing concentration, there is some capacity to non-binding the antibiotics, so they can withstand the action of this. So this can be explaining the not full dependence on beta-lactamase genes. And the most important carbapenem beta-lactamase gene is the CFIA gene. It is causing the wide spectrum against the wide spectrum antibiotic carbapenems to be resistant. It was cloned earlier, and later turned out that it is regulated by an insertion sequence element. Also, we know that the Bacillus fragilis population are separated to two divisions. One is CEPA and CEPA negative, CFIA positive, and the other is CEPA negative, CFIA positive. And it could be differentiated by Mardi-Toff mass spectrometry, and we can detect the IS elements by PCR, and by these we can see that not all highly resistant strains are activated by IS element, but there is some other mechanism that we investigated. So it is mainly mediated by the so-called heterogeneous resistance. So we have an inhibition profile by E-test on the left side, and you can see some resistant colonies inside. So this is the heterogeneous resistance, and the CFIA element is just shown upstairs, and we can induce this heteroresistance by adding carbapenems to very high level. And now we are just thinking that these gnat-and genes, which are acetylases, they could be toxin-antitoxin pairs that could mediate the tolerance first to the antibiotic, and then when the antibiotic is removed, then they can express the resistance. And I mentioned that there is two populations among the Bacillus fragilis strains, CFIA negative and positive. This is division 1 and 2. And the pan-genome comparison just has shown that there are some genes that are specific for both groups. There are group 1 specific genes, and there are group 2 specific genes. But sometimes this CFIA element is characterized that it is not just a simple insertion to the chromosome, but a replacement of a specific gene, and it is valid for the CEPA gene too. So there is some more issues to be researched in this field. And we also had not just clinical strains, but strains from feces, and we found an interesting association between for some genes between the normal flora and the clinical strains. Sometimes it's significantly dropped, sometimes significantly increased, and we hypothesized that a conjugation mechanism can be responsible for that. So more conjugation in the lumen of the gut of the mammalian strains, or less spread in the mucus, and this can cause this difference. And just finally, something interesting. So Bacteroides are characterized for harboring so-called cryptic plasmids that are selfish. They don't code any useful functions, but can be prevalent in 50% of the clinical strains. So one type of this plasmid is so-called PBI143. The genetic structure is depicted in the middle. So they have 3 forms, and American investigators just with examination of metagenomic data, they found that they are also prevalent in metagenomes. They have 3 main types, and interestingly, the industrialized countries the patients in the industrialized countries have more abundance with these plasmids, and non-industrial people in non-industrial countries have very low prevalences for these countries. This picture depicts that, and these plasmids are hominids, so human-specific, and its abundance exceeds the so-called crustophages that was thought once to the most abundant genetic entities in the gut, and now we can just say that 0.1% of the metagenomic reads contain this plasmid, and it was also revealed that it is connection with oxidative stress tolerance and immune tolerance also. So strains with these plasmids can tolerate oxygen more, and in IBD, inflammatory bowel disease patients, these plasmids are more exceeding, so more abundant. So there must be some link between these plasmids and the conjugation of antibiotic resistance genes. So just as a summary, I can say that Bacteroides are both friends and foe. Their biology is intriguing, and there are most probably many unresolved questions concerning their microbiology. And finally, I would thank your kind attention and just show that still there are some connection between Hungarians and Kazakhs. We have Kurgans in the Great Hungarian Plain, and we got some words from the Turkish languages. So with this, I would like to thank you for your kind attention.
Dear Professor Joseph Shoki, thank you for the interesting presentation. So dear colleagues, for questions.
90% is found in north of China, but 25% of it was found in Russia. So Beijing B0, and that's a successful, in brackets, Russian strain, right? And when we made a more thorough study, the line demonstrates the actual boundaries of the former USSR countries in Western Europe and little in Central Asia and Kazakhstan. And here comes statistics, right? So I could make a separate report and STA, and actually the genome data analysis has found evidence to that. So the question is where it has distributed then later on. Starting the 2 or 3 cases of resistance that would raise concerns in our colleagues. Institutes from former USSR, from Russia, and it turns out to be that they would live in Spain for 2 or 3 years, and there was a concern that they could have been, dispersing the disease, and the strains were found, and they actually developed the PCR testing so that they could identify all the most recent cases. However, there were no secondary cases of distribution of those strains. Even though they infected the locals, there was no active TB during that period of time. So actually, those were the short-term summary conclusions made, and ordinary human exchanges, are not enough to introduce and root a new strain among the local population. You gotta have a massive population. You know, when one country occupies the others, you know, the people flee, and you know that strain, you know, the new emergent strain becomes localized. You know, it's not necessarily so that it is totally, you know, absorbed by the local. I mean, in accordance with the local conditions and so on. So the adaptation of the local strains by the local population leads to the situation when the locals become more resistant. You know, that's a hypothetical estimations. And of course, the properties of the tuberculosis strains. And of course, we have to understand that all of the factors are interrelated: virulence, resistance, and prevalence of the strain, human genetics. You know, so actually we can very interesting genotypes. So inside the Beijing, there's a hierarchy structure. You know, there's modern sublines. You know, when we offered that, you know, it was proved that the so-called modern strains, evolutionary, you know, have been globally distributed, you know, and there were not a lot in Russia. And actually, it was important actually to make sure that those strains are more resistant actually. And the two clusters, the diagram of clusters from all around the world, and we had two. And actually, it is more distributed in Omsk region, but those strains are from all over the place, you know, St. Petersburg, Samara, Omsk region, but they all had one combination of mutations, you know, and one of the and those combinations were found in Albania, Poland, and you know, so the combination of mutation was actually useful, and we didn't find the in between. So the other genotype, you know, I called it Buryatia, and it is not as distributed, and it's Buryatia and Omsk region, and they also only have two common mutations. One is the most prevalent, and resistance to tubicin and zonazil, and as I have demonstrated previously, in this case, you know, we don't see any so-called neighbors. So who's closer actually, and in fact it is more than 100 years, you know. So how that genotype was actually, how did that appear in Far East, we don't know. Maybe it came from Korea. We studied virulence of those strains in mice. So we had two parts of the experiment, so the lethality and the virulence experiment. So in terms of the virulence, we would say the bodies, you know, we would kill the mice and after the infection, and then we would watch over for 195 days, you know, lungs, histology, and virus load, and the Buryatia genotype is on the bottom. It was more lethal. Then, for instance, to your right, you could see the modern strains, you know, to include Central Asia outbreak on B0. However, Buryatia became the most virulent and most contagious. So when we looked at the genomics, we found some of the mutations typical for that cluster, to include genes that are responsible for virulence, for adaptation, and in terms of quantity and mutations, all of those mutations were put into the table, and they could have had the considerable impact upon the structure of proteins. And I actually mentioned some of the genes in bold so that you could have a closer look at them. And in terms of the geographic correlations, low virulent. However, Mark Blue, we had the genotype was thought to be a scientific council. So I'm very pleased to work together with you and have a chance to come to certain conclusions and make this presentation and present it for your attention. So over here are 3 million patients who have that disease. And actually you could see the data in regards to Kazakhstan. On the top you could see the graph with the breakdown in terms of the regions. So if you're interested, you could have it. So 11.4% of cancerous diseases is the lung cancer. And lately, for 10 years in a row, the number of registered cases who have lung cancer in Kazakhstan, lung cancer is the problem not only for Kazakhstan but also worldwide as well. And let me not mention about the other factors. There's information, genetic factor, for instance, and other lung diseases risk. There's a prior regulation, environmental, tobacco exposure, and so on. So this leads to the fact that we get lung cancer patients. So talking about radon and asbestos, uranium actually gets as a production of uranium, and if you've noticed from the report of those, he mentioned about the oxygen species growth, and this leads to oxidation stress, and this actually leads to cancer. So. And those radon products actually lead to the destroying of the DNA structure, and this actually leads to chromosome breakdown, and then it also leads to lung cancer. So the hyperventilation leads to changes in microRNA expressions. So there's high levels of oncomarkers and oncosuppression, and then eventually we have cancers to include lung cancer. Well, talking about asbestos, you could see that there's ventilated cells engaged, and fibroplasty and isochemo cells, and then the same, asbestos, fibrosis, and then the cancer. So you could see that those two cancers are very strong factors, very considerable factors that lead to the breakage of cell structures that lead to cancers, to include lung cancer. There was a question, I know that there are not many experts, but we have those who deal with medicine and molecular medicine and molecular studies. There are lots of diagnostics methods for lung cancer patients, but we have to find an approach in order for it to be non-invasive, right? And we made an offer. OncoMarker microRNA could be a potential biomarker of lung cancer. So together with Professor Isotta, American, so we made extracellular microRNA in a liquid biopsy, applicability in cancer diagnosis and prevention. So that publication was made back in 2016. So we looked at the entire group of cancerous diseases. That article actually is well cited up until now. And over here you could see microRNA. And there could be microRNA that generate, that lead to cancer, but there are some that are participating in a normal genesis, invasion, and metastasis. All of those factors tell us about the interrelation. MicroRNA actually is a small molecule that is consistent of roughly 20 nucleotides, and it actually participates in the acute process generations. And the microRNAs actually could be fixed not only while having the cancerous diseases progressing, but also in other processes as well. So microRNA and radon-induced lung cancer, is demonstrated on the screen. So it depends on the dosage, on the concentration of radon in the human body, and there's a direct correlation in between how much radon is contained in the human body to the chances of having cancerous diseases and having more or less expressions of microRNAs. So the green is microRNA profile in higher concentrations, and in between you could see microRNAs 146 and 147. So in both cases, they do get registered. So the error to the going up and going down, could actually lead to the decline or to the decrease in terms of the tumorous regrowth, right? And here comes the question, right? It would be nice to study those processes, and the purpose of the study is to search for molecular biomarkers of lung cancer. So over here you could see a different type, 125 and 320C in blood plasma, and you could see the clustergram, and as I said to you before, different types of RNAs, they have different effect, and they could be actually clustered and grouped in terms of the effect that they have on the spectrogram. And so 720 micrograms was tested. Those, he was mentioning about mitochondrial effects, and mitochondrial effects actually could be so that on the top you could see actually that with the help of those receptors we could get eventually alpha and beta pathways generated, and then after that this could impact upon concentration of immune cells that regulate immunity, and then later on the inflammation is also the case, and the next step forward, and then mitochondrial DNA, RNA, and it leads to the cell formation, and then inflammation leads to spaces, and this is a part of lactose processes and so on. So eventually all we have is the pathogenic flora. So asbestos increases production of, and as a result of oxidative, cellular stress, You could see the results demonstrated on the screen, and we could see the active form of oxygen that was studied with the fluorescent spectrometry. And different concentrations were compared, as well as the longevity was studied. Toxic effects. So actually, in most cases, single and double-strand DNA breaks are caused by ROS, and actually, this is the result of the oxidative cellular stress. And actually, we tested, and you could see the comparison to the control group, and different dosage demonstrate different results, and you could see perfectly well here that those who are interested in dealing with the process know perfectly well that destroyed DNA is something that would get in a eventually, cytotoxic effect of the life expectancy of patients. So actually, we have published these papers in scientific journals. So I would like to thank you. Thank you for your attention. So I'd like to thank Ms. Kusainova. Asa actually made it through the University of Genoa, and she has recently defended her thesis there together with the Professor Yezuiti. So we actually educate not only here at Eurasian University, but also in Italy, and Olga Bulgakova, Kusainova, all of our young scientists, made it successfully to master degree programs and doctorate degree programs. So once again, I'd like to say that's a great chance for me to speak. We have lots of urine, and where uranium is, radon is, and the Ministry of Ecology information to make the measurements, because radon is found in all of the households, and what we do is we make the measurements ourselves, and then we compare the data and have them compared to whatever data the Center of Oncology have. And then we interview the patients and work with the medical papers. I would like to say that this is a very complex work, to find such patients. We are not the doctors, we are biologists. It's hard to do, so we found ourselves in a pretty complex situation, so maybe we need to work with the plants, although I used to be working with plants while being at the Kazakh National University, so perhaps we would need to shift, to collect blood is extremely hard to do. Thank you so much. Any more questions? No questions. Thank you so much. Thank you so much and good luck. Thank you so much for such an interesting presentation. Thank you for moderation of the wonderful session. So our first part that was supposed to be provided before the lunch is over. So we have two announcements. So we're pretty hungry. So lunch is going to be served on floor number 1 and floor number 5. So right after, we would like you to join, but before that we would like to make a family photo that is going to be made at the entrance to the main building. So please take the photo. Those who are not going to be on the photo won't get the lunch, I'm just kidding. So actually, please, let's get together and make this photo. And we're going to be having a nice poster session. And the author of the best poster is going to be made. So the commission is Mr. Margousov. and Mr. Dr. Baeva. So they will be actually our jury members, and they will select the best poster. So from 1400 to 1500, and plus we're going to be having the online voting for the best company of the exhibition. So you know that the stands were prepared. So if you think that the company is the best, just you have to approach the QR code. So approach the columns, have it scanned, and vote for the company and the stand that actually brought your attention. So let's make the family photo on the first floor, on a staircase, and then enjoy lunch. Thank you. The channels of detection. So this is the optic scheme of sight. So 3 types of lasers are being used, and the 12 channels. And you could use it in order to analyze not only the cell of an animal or a human being, but also the cells of plants, bacteria, algae, so anything. And over here you could see different examples in terms of why and how you could use this machine, you know. For instance, labeled Zamazon particles absorption, and this is a very detailed spectrometry and a wide range of images, and with the help of the flow cytometry, you could actually make the records of 20,000 images or 100,000 images, and then you get the quantities of Zimazan as it is depicted here. You know, the first channel to the left is the light spectrum, and then the combined image, for instance, endocrine cell, you know. So that's one of the examples of how you could use those machines. Or other, so dendritic cells, you know, we use antibodies or the RBD domain of the 5 protein, and this is SARS-CoV-2 virus absorbed on aluminum hydroxide, and this was visualized, so we could actually use the prototype vaccines, and this is how you could actually assess the protection. So actually the dendritic cells actually could be analyzed with this type of cytometry, and those are the monoclonal antibodies. So on the spectrum analysis, you know, we almost see nothing, but using the fluorescent machines, we could see those points, you know, which are lighted, and you know, this is something that you could find on the screen. And intercellular interactions, you know, it's easy to pronounce, you know, and actually the non-cultivation of CD209 plus monocytic dendritic cells and CD3 plus T cells, and assessment of cell inviolability, you know. We could also not look at the intensity of capturing, right, of dying agent, but also, you know, to look at the acids. So this is the reference to 7-AAD. So you could see the dying agent. So visualization of the core, you know, so any zones of fluorescent microscopy can be used, you know, so cyto and cyto deep red. So examination of the T cell proliferation, the same proliferation in this reference demonstrates of how to use a target valid. So very interesting analysis, which is something that we plan to do. So those are the pooling particles at the HCV. You know, you could see the birch tree analysis. Thank you, colleagues. There's going to be a conference in November devoted to molecular biology, so please participate and include the format of hybrid. Thank you. As we have agreed, please, the next presenter and strategy of having natural-like medicines of the new generation through the prism of microbiome. I would like to thank the organizational committee for the invitation to make my presentation. So just a couple of minutes in regards to the anniversary. So we've been celebrating the 30th anniversary, and our institution, Institute of Genetics, has been actively engaged in forming and developing the fundamentals that contributed to the establishment of this institute in Stepnogorsk, and many scientists would actually interact and work with our Kazakhstani colleagues back then 30 plus years ago. And it's great that we are having now a positive result represented by this institute, by the equipment and the people. And today I just wanted to be briefly telling you about the new, relatively new area which was established 10 to 12 years ago. The mainstream in science is 5 to 7 years, and when it lasts for 12 plus years, this is a stable mainstream, This is a new interdisciplinary science that has been actively developing, and actually this is something that we expect a lot from. So what have we been expecting from this area in particular? Sticks. And you could use it as an ingredient in food supplements. And microbiome is directed products. Those are the new definitions, and yes, indeed. Whatever we have just started discussing and using different conferences to include the Council Federation and Duma. What is it actually? Microbiome is something that could be tested actually, and this could lead to some positive properties while dealing with certain diseases in human beings. And I will talk just about some of the labs that deal with this here. We talk about live biotherapeutic drugs based on lactobacilli and postbiotics, metabolotics, and components of probiotic bacteria. We've established efficacy, safety, and technological applicability for industrial production. For instance, Lactobacillus brevis 47 while having radiation or chemo, and perhaps while correcting some violations in terms of immune performance, to include Cartier. And of course, proteins and enzymes and low molecular weight metabolites with an established mechanism of action. It is almost the medicine and has to be in compliance with the requirements for medicine. So this is going to be protein FM3. Which could be selectively related to other cytokines and regulate the cytokine storms, for instance, and so on. And why do we do that? We do this having acknowledged the national program in Russia, and I will close this program, and we do realize real groups of population will be consuming those innovative products, and actually this is a purpose for healthy population and those working in stressful conditions and levels of radiation, for instance, those who live next to radon and uranium, and those who have permanent low temporarily in ecologically unfavorable regions, a group of people with nutritional imbalances, not for us, but for instance, the African countries, and groups of the population that require adaptation to new living and eating conditions for them, shift workers, students of large megacities who came from different regions. And definitely we target socially significant categories of consumers of innovative products, neurological diseases, depression, autism, and others, neurodegenerative diseases, Parkinsonism, Alzheimer's disease, removing the effects of chemotherapy and radiation therapy, mucositis, immune disorders, and others. Antidotes for low-intensity radiation exposure, waxy rehabilitation of certain population groups, and COVID is still there, and post-COVID syndrome is still with us, so we have to somehow actually work on that and provide a support has impacts, both positive and negative, So whatever depends from microbiome is, and what is that? The studies that look at the mental health and cognitive capabilities, how is it possible to make corrections in terms of in terms of getting the exposure to microbiome and
to correct the epigenetic regulation of various programs of human brain functioning. As everything in this world throughout the last several years, microbiome being digitalized, characterized as well, and that helps us to formulate the algorithm of validation of biomarkers and using them to characterize microbiome in common condition and in different pathologies. We started from the disease stage and so on. The formulated algorithms in U21 relieving the effects of oxidative stress in neurological diseases. We preventative measure of Parkinson's disease. With the use of new technologies, we identify the genes and enzymes which works within the anti-inflammatory processes in the body. They depict the positive results in animal tests on curing some side effects of Parkinson's disease. First of all, as it's been mentioned, it affects the anti-inflammatory processes. It helps in terms of stress disorders and so on. Here we can find a set of examples. Beyond this, everything been possible to implement with the use of complex approach. We conducted some set of analysis and tests. Initially, it's been depicted that for Parkinson's disease and other diseases, it has certain features which helps to account it as the medicine. We used the method of genome analysis, decreasing the set of ingesting time, increasing the microbiome transcriptology approach, and to identify the genes potentially could affect the treatment process of Parkinson's disease. With the use of following genetical evidence, we've seen that on molecular biological strains, it really works and it can affect the situation. In the National Center of Biotechnology, I've seen that you have a long history of firstly depicted how the bacteria could be synthesized And the key advantage of this approach is using the relevant number of bacteria in order to make it synthesized, and using additional mechanisms like azotol could be as a kind of a protein, bifidobacterium protein, as we've depicted, could selectively connect with cytosines and work as an anti-inflammatory agent. We can use it for which work in collaboration with industrial partners and governments. Direction moving forward with the pharmaceutical industry. We design also the functional products. We're trying to move forward using the modern approaches, bring us the new breakthroughs, and we'll be proud of their attention on some kind of technological work. Could you please switch on the slides? Including the isothermic amplification of RNA DNA. In details, we would like to discuss the tools for its realization. Isothermic amplification of DNA is a kind of an amplification which is run on the stable temperature. Actually, it doesn't require the thermal
enzyme stated was special SIMSA 777. That strain actually has an interesting history. I don't know if you remember about the protein called 777. Actually, that enzyme was identified from the leftovers of that drink, and actually was expressed and was good in terms of the performance. And plus,
он улучшает прохождение амплификации длинных фрагментов. Мы видим здесь продемонстрировано для 4-8 килобайт. Ну и
we do have
некоторых достаточно злобных ингибиторов,
and there were the strong inhibitors that we use in clinical practice, and we use this in order to get Humira-like enzymes. And actually, by the time when we started working, we would have it for classic DNA necessary quantities, this makes us, be sure that this could be actions. And actually, I wanted demonstrate what we were doing afterwards with the enzymes. And that work was really interesting, and actually that is the golden standard. The hybrid polymerase, compared to the other properties, actually demonstrates that for our enzymes we could have this 2,0 positioned as the core for such sort of tests. And our foreign colleagues actually were very effective in terms of using hybrid revertase, and I told you that it has pretty unique processing. Set the timer for 14 minutes. If you can hear it, this means that you have one minute. So please make it. We're behind one presentation. Dear colleagues, I want to congratulate you with the 30th anniversary, and actually I assume that 3 or 5 years later we're going to be having new developments that are going to be actually substance with the UF spectrums and the nano sizes. So it is 44 nanometers. And actually, we have also developed for the first time ever worldwide. We came up with the method allowing us to buy the quantities of biological liquids. And this slide actually demonstrates that the biological accessibility, we use dogs for studies, and we use the same dogs in order to look at the cytochromes, in order so that the metabolism remains stable, and compared to the original component, supermolecular complex was 205 in that position in particular tableau, and we made the in vitro studies using the cells of melanoma B16, and we came to that. So we actually compared those cells, and I told you about that component not being able to get solved in the water. So, and next cells were taken from the patients who do have the breast cancer after incubation. So depending on the incubation period, we could see the deviations and very strong curve demonstrating the dynamics in terms of growth, and actually it was shrinked. So here comes the typical cellular culture, and as we could see it on the screen, cells and this inhibited growth, and what conclusions can we make in terms of in vitro studies? We can see that the growth is slowed down thanks to cytostatic action during day 3. And on day 5, thanks to the toxic effect. And those effects were found in a spectrum of 10 to 12 nanometers. We know about the in vitro studies that you can, get splitted while we're shifting over to in vivo studies, and the effect could be demonstrated in vivo studies. So over here, what we did, we had the preventive activity, and actually, in regards to the cancerogenesis was provided by diacetylnitrazolone, and we would have rats getting this during a long time, and the animals were splitted. The male rats were actually formed 16 animals in each group, and in control groups we would not get the medications, and with the concentration in the quantity of 25 and 50 milligrams per kilogram. And here comes the results, 10 times less tumor formation in the liver, so that was the prevention effect that I mentioned about. And the same was made in regards to the clinical studies. We made the studies. And we used the carcinoma lures in lung cancer, and the results actually was adjusted in accordance with the dosage demonstrated. And we can see at the moment that the deviations, so the suspension in growth was really considerable. However, in accordance with the requirements, it was up to 90%. However, we couldn't make it, we just made it up to 70%. And then we made the analysis of cell growth, and we received great results. And we identified the quantity of metastasis in the lungs, and in terms of inhibiting metastasis, that data was in compliance with the requirements, and there was a considerable decrease in the table. So the studies of the same component actually shows us the impact upon the biochemical performance, and since this is the inhibitor of glucocan civil and we watched over the protein exchanges. So actually it has no impact upon antioxidant system, and actually it impacts through superacidity of but at the same time, you know, when it comes to and it was the first time ever how this would impact upon the hormone status of the animals, and actually that same substance was no impact upon the major hormones, and you could find them in the table. So we took the diabetes mellitus, and what we saw was that the levels in glucose actually, and that's actually rarely happening, that when the animals with hyperglycemia would have absolutely normal sugar rates after the anti-cancerous treatment. So it was almost a norm. And in case if we deal with the diabetic patients, then that ratio would normally be up to 15 units. So it normalizes the metabolism, and you could see it on this table. And next, we made the glucose tolerant test in bunnies, and the level of sugars also went down, and we could see on the curves on the x-axis that it takes up to 300 minutes for the sugar rates to go down if the medication is effective. So the question is how safe it is. So we made the the studies in bunnies. We took the swine, and we came to the conclusions that toxicity in case of one single time use refers to class number 5, and LDP it was hard to identify in case of oral intake. And then later, in case of the chronic toxicity was not found, the substance was acknowledged as safe. We made the analysis of chromosomes, and over here, this graph, you could see that the control and those who are getting it, the level is almost the same, and the positive control was at the level of 20%. Thank you very much for your attention. I think I made it. Next report, Mr. Shustov. So he's going to be talking to us about implementation of I work for the National Center for Biotechnology, and my objective is to tell you how our center and our partners from CAR-T therapy in Kazakhstan. So CAR-T, what is it? So I would like to say that we don't have enough time, because let's finish by 5:10. By 5 o'clock, I'm not going to be talking about CAR-T, and so CAR-T is an adaptive cellular immune therapy, And actually, this is the personalized medicine. So cellular product actually is made individually for each and every patient, and in essence, CAR-T is effective to patients to whom conventional treatments have been ineffective, who relapsed of refractory diseases. So how do we do CAR-T? Cellular therapy, so the clinical stages. So patient gets leukapheresis, goes through leukapheresis, and then from leukapheresis they separate T cells, 4+ and 8+. They make the genetic modification, and the essence. This is the difference in CAR-T between the other cellular therapy, and once the quantity is maximized, then it gets back to the patient. CAR-T, as I said to you, is effective therapy, and you could remember one figure in 2024 this year. The number of CAR-T worldwide achieved 500,000 people. So this year, 0.5 million people worldwide received CAR-T. Why? Because it became a really important approach. Unfortunately, in Central Asia, it's not many. In Russia, it's only 2 clinics. Only in Rogachev Center and Hematology Center. These studies were published, and in Kazakhstan, CAR-T never was used. At least 3 Kazakhstanis received CAR-T abroad. And this is the illustrative slide demonstrating the Humira-like antigen. So this is the artificial protein. So the essence of CAR-T is the method allowing the immune system to target pathological populations. So immune cells get notified by CAR, giving the cells new immune characteristics and properties. So actually, the receptors are of different specificity, right? And after the genome modification, the cells do have the similar immunological specifics, and they then attack the pathological cells. And actually, there's a single core cell and dual core cell. One core cell can attack and destroy actually 100,000 of cells. So overall, we can get rid of a kilogram of cancerous cells. At the moment, CAR-T can be used only for very B cell B-cell cancers is CD19 and BCMA antigen cancer. And this is something that we could use against lymphoma, melanoma. And what is the problem? The CAR-T therapy is still too expensive for developing countries. It's very, very extremely expensive. The prices of registered therapies, it's $475,000 per patient, and the producers don't have a program for emerging markets and for having prices going down to these countries. So the objective of having CAR-T in Kazakhstan is related to this fact. Actually, we need just not to arrange the production and we need to make the therapy accessible in terms of pricing for the Kazakh citizens. Just to compare, the most expensive reimbursement is roughly $30,000-$35,000. So we have to target the spectrum of prices.
Я говорил, что
I told you that CAR-T is effective, so what do I mean under efficiency? You know, worldwide the CAR-T is the last line of therapy. It's only for those who would not have an effect from traditional conventional therapy, and they don't have a large choices, right? It's just CAR-T, in fact. And after the application of CAR-T, in 90% after the first months, during the inpatient clinic is full response, so the disappearance of cancerous blasts and the efficiency of CAR-T is different, dependent on the dosage, dependent on different clinical forms of cancer. And if you're interested, so it is 50 to 80% of people who would have full response during inpatient period. It is going to last for a year, and on average 60% of those who had CAR-T and those who had full response during inpatient period would live 5 years without any recidive. So for 5 years they won't have cancer coming back. And you know, they survived 5+ years. You know, they are considered as those who became healthy. You know, I remind you that those are those who were not getting any effect from the conventional treatment. CAR-T is not safe, but the mechanism of this therapy actually considerably impacts upon the immune system, and the immune system is getting destroyed by cytokine storms. You know, it's neurotoxic and so on. So why am I telling you about that? This is a very complex therapy, and we need not only the biological base, but those who are going to be making the product, but we need to have prepared physicians, because CAR-T never was used in Kazakhstan, and we don't have trained doctors to have CAR-T in Kazakhstan. So what else can we say? We speak about those people who, after all the treatment lines, have been, you know, suffering a lot, and there are other risks. For instance, the immune system in such patients is so destroyed that there is a risk of having potential bacterial virus-related diseases. Of course, the treatment is to be provided in an inpatient clinic with the trained doctors. And what are the conditions for this therapy in Kazakhstan? You know, biotechnology base. Our center, National Center for Biotechnology, is going to be making and producing the cellular CAR-T products. And the Ministry of Healthcare is planning to start funding in the end of this year. Plus, we need to have the clinical base, we need to have physicians, plus the material basis and funding in order to fight the negative consequences of the therapy. And those medications have to be ready to go, and plus the materials to make the product. It's very complex, you know. We have lots of materials to be purchased, you know. However, there are issues, you know, and I will tell you about one of the issues, which is the accessibility of reactor. And what could be the condition for CAR-T in Kazakhstan? I mean, the legal framework. And as you know, the products may be used in case if they are registered. If not registered, then within the clinical trials or as clinical exceptions, inpatient exceptions. So far, not a single global producer of CAR-T didn't enter Kazakhstani market. CAR-T is not registered in Kazakhstan. The only chance to use this product and such therapy in accordance with the hospital exception actually shrinks the list of clinics and hospitals that will be able to open the doors for CAR-T. And actually, in fact, from the oncological centers, in fact, it's only one, which is the National Center of Oncology, who are the partners within the framework of the program with us.
So what else would I mention? So I told you that the difference of CAR-T from other types of cellular therapy is that we use gene-modified cells, and all CAR-T therapies are purposed for genetic modification in order to have a CAR. Gene is transduction with heterovirus vectors shifting, and we don't want to invent anything. In terms of the therapy, the program is purposed not to invent new technologies, but to introduce whatever is existent. And we want to use retrovirus transduction. And in fact, you know, the lack of accessibility of lentivirus products, you know, you can't just purchase them simply, you know, you can't just you know, pick it from the list of purchases. You know, this is not how it works in this industry. You know, the vectors for CAR-T, well, you know, the Novartis or Generat, you know, they don't sell it as vectors, you know, standalone products. You know, you could actually purchase this, the service actually of getting it, but this is going to be your own responsibility. You have to make the structure of this vector, you know, for clinical application. You cannot just use any lentivirus vector, but you know, we shouldn't be compromising the technology. We have to be make sure that there is a variability. So there has to be the GMP certificate and the certificate made by the producers. It has to be either as a part of the registered therapy or hospital exceptions, and those vectors won't be able to be just purchased all over the market. Moreover, you know, what we've been leaning on, we do have the equipment for CAR-T. So you have Clinimax Prodigy. This is the industrial standard for these products, and canal therapy is Novartis that has 2 plants, one in Germany, one in the United States, and in Kazakhstan we have one here in the center. So this is how it is working. This is the separation of electrophoresis and the production of the product. So this is how the CAR-T cells look like. This is how they get grown in the incubation camera. So actually we make the control of quality of the final products with the flow cytometry, and the equipment that we use in order to have the population control are well known, and you know, we're not inventing anything. We're using standard tests, and whatever you could see on the slide is that in the cells that we have received, almost all of the cells are lymphocytes, and in fact, actually, in terms of the cellular performance, this product could be used and should be used for the human beings as therapy medicine. So actually. So currently, National Center for Biotechnology is in the process of creating clean rooms at its site to accommodate the production of CAR-T cell products, and we've been dealing with the alternative options. You know, I want to have it very clear. CAR-T is very complex, and to have it just leaning on your own capabilities accessible for the Kazakh experts is not possible. That methodology was registered back in 2017, but it was the first CAR cells were received 40 years ago, so it took 40 years to make it through. We need undoubtedly to have the foreign partners in order to transfer the technology. We do have the clinical base, and we have one partner abroad, and we've been looking
for partners who would be responsible for transfer of technology to Russia and China. That's all that I wanted to report. Thank you. So what should we do? Let's have one more report. And so Anton, CAR-T cellular therapy, so from studies to production. Good afternoon, dear colleagues. Well, in fact, from day one of the existence, we've been partnering with the National Center for Biotechnology, and from the establishment, we've been partnering with them, and we provided the first genetic analyzer and the first ever machine for the mass parallel sequencing, and we would like NCB to be free of any limitations and let you be funded and supported by your government so that all of your hypotheses get approved, so for you to have more money for your studies. So my congratulations with the 30th anniversary. So in fact, while getting myself prepared, I thought that I would actually sort of foundation, and then Alexander Vyacheslaevich is going to be telling about the results. In fact, the results do exist. It's the first cellular product in Kazakhstan, however, with the approved phenotype. And in my presentation, I'm going to be summing up about what cellular therapy is. So using the cellular product, using cells, is purposed in order to have allogenic donor cells. That would be nice to have one donor, to have white cell bloods and white cells, and have it added into the blood product. But the immune system is very complex. We have a lot of pathological reactions, and so far it is autological. So actually, this creates a lot of complexities. And at the same time, at the market, there's a lot organizations that speculate in cellular therapy, and as a commercial product, they make advertisements of stem cells and so on with something that is not in evidence, and actually this blows an interest and trust in such therapy. So actually, in fact, Alexander told you about what CAR-T is. I'm not going to be elaborating more on that. I will just reiterate that the first patient, Emily Whitehead, and she is alive, and she's in remission for 10 years in a row. I've heard this girl's dad is very active. He's been promoting CAR-T, and that CAR-T has been registered as irregular terms of products, and it was approved by FDA, and she participated in cellular therapy, and Zalma officially actually distributes that product in Kazakhstan, and Thermo Fisher participated from day one in terms of creating, for instance, cellular products, or but the magnetic particles that are used, because there's a parcel of white cells, and in order to have them, separated, and to have T cells, in order for those that were able to get, modified, this we need to have the magnetic particles. So actually, you gotta have 4-CD antibodies allowing T white cells to get separated. So actually, that magnetic agent is used in therapy of Tumor, and Yescarta and CART Thermo Fisher scientific, the environments is not permanent, and you gotta use some replacements of the immune mixtures with the constant chemical content, so that there's safe to have the transfusions, and you gotta use the special agents and environments that have brain CTS. CTS is a special product line. It's called CTS Optimizer. So all of the substances are artificial substances that have been received chemically, and the chemical content is known, and they've been certified by GMP, and they've been traced, and this is something that you need for the therapy. And majority of the product actually uses the agents of Thermo Fisher Scientific. However, I have to reiterate that while working on this slide, the studies are conducted by Nazarbayev University, and actually objective is to use a solid tumors with this product, and in fact cellular product is good to go in terms of solid tumors, and it can attack from external space. And actually we need to use the same cellular product, and the company that you see, Pact Pharma, actually in its studies have mentioned about not transforming the cells thanks to lentivirus. As a lentivirus is a pretty dangerous thing, and they use adenovirus vectors. So CRISPR-Cas9 capable of giving the ruptures in the given places with the genetic constructions, and in order to make the genetic editing works, they don't use the virus, but they use the flow-based volume. And yes, in fact, that technology works, and it was successfully in order to treat solid tumors, and the special machine was used in order to have the cell therapy system. So that was the branding. So in fact, there are lots of products made by that company, and they've been commercialized, and those products were used in validated FDA-approved therapies, and some of them are being used at the clinical trials. And I need to say about using non-traditional approach, while they put a cellular product, actually the traditional approach, is something that while you're using a conventional approach, laminars, cell boxes, and standard centrifuge, pads, and so on. But in order to have cellular products, you need to have 2,000 different premises. But if you use the automatic Prodigy and Miltenyi, you would need twice as less area. But Clinimax Prodigy, has one malfunction. The processing takes weeks, and in some processes for 2 weeks. Can you imagine that such an expensive system is busy? But we have 299 patients. 200. These systems have fixed productivity per year. So large centers leave to the automation of the process, and they have, production of enzymes, for instance, and they grow not in Clinimax Prodigy, but in incubators where you have more capabilities to grow the cells. So Thermo Fisher, again, is something that suggests, those are the module-like systems. Those are the separate systems. One is to clean the cells, one is to absorb them, to pump them, and so on. And for these systems, you need less area, less maintenance personnel members, and less clean labs and so on. So
Those are the module-like systems. You can put them together physically, and you could actually use different software, enabling you to control the cellular processes, getting the product prepared. So let me comment on the systems separately. We produce systems to work with the viruses or to work with the protein.
The virus particles for decapsulation in the Republic of Kazakhstan have been successfully used in the National Center. Dr. Zabuna for the bacillovirus production in research in the vaccine. In fact, for immunovaccination. We use not the common type of vaccine, we use the lentivirus and adenovirus systems. And with the help of the feature, we can build a box using the lentivirus, and all of you need to do is to adjust the construction tool in order to prepare the adjusted environment. Part of that, there is a need on adjusting the vectors of lentiviruses. So lentivirus vectors are much more popular in comparison with common ones, and they use the box samplings and secretion of other components in order to make your virus in a different design. In terms of preparation therapy of lentivirus, it is quite long in comparison with common one. It takes 1 or 2 weeks for creation, and there are some means in terms of decreasing this duration. We have systems which help us to release the particles from cells that help us to decrease in time of production and increase the quality of the product. And the costs of therapy are quite high due to the long period of incubation of the cells and tracking of their growth. Probably they losing their potential due to the fact that we get the cells, transforming them, and then they take 7 or 14 days for growing, losing their key features. So they got more markers of differentiations, and that makes it less effective. Apart of that, special magnet particles been used for research, changing the approach. They take the particles, accentualize them, and hold them together. Then they connect the cells. The approach of connecting the cells requires the lentiviruses use in order to make it a chimeric one. So we got the genetically modified cells, and so we use this moment for adjusting the speed and effectiveness. We use the magnetic cells in the buffer system. We can freeze them immediately and send to the clinical institutions for the further work. Last year we presented that the process of this product for the cell therapy could be reduced to the 2 days in total. They use the advancedly prepared cellular product that helps to use the time efficiently, and part of that using the 1 day for the complete preparation. For the product preparation in most centers of cellular therapy, they do not use all-in-one products, but the tools for separate stages. There is an interesting system for these stages, like a centrifugation system. We can send the fluid for simplifying these cells. It depends actually on the situation. For instance, we have a blood rich of red blood cells. We separate red blood cells with and white blood cells, make the solution of a concentrated white blood cells, and these systems allow us to make universal and diverse operations of manipulations involving cells. This approach is used by the large enterprises in order to make more product per time. The following system I would like to demonstrate. It's the flow cytometers with visualization. There's been mentioned some necessary information by previous speakers. Our tool mechanism called Attune PyX mechanism. Actually, the product we got with the help of it is quite unique. You can not only see the sarcoma ramus cells, you also can see the interaction of cells on the graphics. As an example, you can take a look here. We can also decentralize them and see the potential use of the cells. And it's been proved also by the use of Thermo Fisher apparatus that the cells can attack cancerous cells as a steroids and destroy them. We can use the automatic apparatus for biometrics, and we have the complete portfolio for the cellular work, for the cellular therapy, modular therapy, and we have running research works here in Kazakhstan in collaboration with Kazakhstan teams and foreign groups. In conclusion, I would like to mention that we have a competition for our clients. If you scan the QR code for subscription in Instagram and then fulfill the survey, you will receive some free genetic tests, and one of them is the origin test. Thank you for your attention. It seems like we finished with the work we plan to have before the coffee break, and probably we will reduce the coffee break time up to 10 minutes. So please take a cup of coffee with you to take a seat here in the hall. Thank you. CAO strain. The key target of our work being to conduct the molecular genetic monitoring of the population, tuberculosis population in Omsk region. That is why we investigated the strains of 2021, 2022, worked together with patients. We analyzed the collection, identified, separated from the Актовый region, necessary data, and 2008. Here you can find the methods we used in order to subtype the genotype among the patients with tuberculosis. We've seen the indicators of international statistics. As it's been noticed, the major part is for men. In terms of age, from 31 till 75 years old, and in major parts they have the genotype tuberculosis of lungs. In terms of phenotype, more than half of strains has the resistance to the medications. That is the major parts of the common medications, and around the 16% has the resistance to the ticanolone group. In terms of new terminology of WHO, it has been featured with the high level of resistance. According to data from 2015 till 2016, there is no relevant distinguishing factors. Here you can find information on structural population of tuberculosis in Omsk region. More than 66% of strains been devoted to the Beijing, Eastern Asian phylogenical line of strains, including B0 and BW48. Other strains belonging to the other families, like Euro-American line L4, and throughout the 5 years period, there is decrease in tuberculosis, ancient sublimation of tuberculosis been decreased by 2 times. The other parts are not relevant and remains at the same amount. Here you can see the correlation of different strains and belonging to the different genotypes, and it is clearly seen that more than 77% belong to the Beijing genotype, but there is some difference between the genotypes of Beijing and CAO. There is a difference at the percentage level, including 82.5% and 30.3%. If you will take a close look at the central CAO, typical for Central Asia, you will find out that there is a rate of 8 out of 9 strains, and the ancient subline has the feature as drug resistance. It has been stated that the Omsk region has a long border with Kazakhstan. It was quite interesting for us to correlate the data in Kazakhstan and in Russia. We've used the publication of Ms. Akhmetova, published in 2022, and it is seen that proportions in Kazakhstan and Omsk region are quite relative. And if you will take a closer look on the subtypes, the patients, we have 6 times more patients in Russian, in Omsk region, due to the high level of resistance. In Kazakhstan, around the 46% of resistance. In terms of Central Asian subline, in Kazakhstan, Republic of Kazakhstan, we have more drug resilience in comparison with Omsk region. Unfortunately, this work hasn't covered the CAO subtype. That is why we discussed the key findings of other work from Kazakhstan under the governance of our colleague. We got the collection from 2008 from the north regions, including Akmal region, and we've used the same methods, and among the dominated genotypes of Beijing, there is a dominant ratio of CAO genotype, including 60% of multiple drug resistance. We also included into our analysis the key findings of our colleagues from your center. In Astana, they gathered the data in 2021. They identified that among the strains, they got more than 60% on CAO strain. So in fact, we have the huge clinical and epidemiological relevance for the Kazakhstan Republic. To conclude all the information, I would like to state points briefly. In Omsk region, bordered with the Kazakhstan, has more samples of Eastern Asia strain of L2 and Euro-American phylogenical lines. The proportion of Central Asia outbreak, Beijing in Omsk region, is still not relevant, reaching the threshold of 4% in comparison with complete domination of the strain in Kazakhstan with the proportion of 64%, and that plays a huge role in terms of drug resistance. Thank you. Thank you. The next presentation would be online, the new vaccine Pollenvax for the treatment of allergic rhinitis. Do you hear me? Yep, the sound is clear. There is no access yet. Today we will have the presentation online. That is why if we will have any types of technical issue, I would like to say sorry in advance. So dear colleagues, I would like to congratulate the National Center of Biotechnology with its Jubilee and wish the best for you in terms of a key topic. It's been announced already. I would like to cover one of the global issue we have today connected with the disease that in rare cases has a lethal case, and it impacts a lot the quality of life. The disease is named allergy. When I mention the quality of life, that includes different allergic reactions, including the most spread, that is allergic rhinitis. That is a season disease for flourishing plants that accompanies with the coughing, sneezing, and during the springtime that decreases the productivity level, and the quality of life is deteriorated in general. And that is a common issue for the wide range of people, and the key target of this picture is to depict the market in this field in regard of the preventative measures. We are waiting of the increase in these indicators in general?
So this is going to be thanks to the Asia Pacific to include Kazakhstan. And currently there are two methods of treatment. Allergic specific immune therapy, and you are well aware about other types, which is symptom based, corticosteroids, antihistamine medications, and this actually is something that is treating the symptoms, but it doesn't cure the reason. And the golden standard over 100 years ago, it was actually offered immune-specific therapy, and this is what we do. We ensure that there is immune tolerance to the disease, and nowadays there's sublingual or transcutaneous and the transcutaneous method is the most widely spread method of treatment in rhinitis. Why are we dealing with it globally and locally? So actually, the grass known as Jusan, there's including pollen wax. The wormwood is the most allergic grass, and actually, this is #1 for Kazakhstan. And why is it so? So what is the problem? The problem is that whatever we use allergic-specific immune therapy has a long-term course of treatment. Minimum 2.5 months up to 8 months of treatment. And plus, on top of what I just said, in order to have the results fixed, you have to have it repeated at least 3 years in a row. So what is the problem? During the treatment period, there's lots of injections, and sometimes, it has the compliance issue, right, when patients are not prone for treatment, right, and they actually finish it beforehand, and what could be the solution, actually? This could be the solution that is able to be used, in our regard. So choosing the vaccine-specific antigen and recombinated protein, and we picked it as it is conventional, is presented in all types of wormwood, and 95% of patients who are sensitive to that allergic component react to it, and it is less allergic thanks to localization, and actually, in fact, it is less allergic, and it is good in order to have the vaccine created. Actually, should we talk about the transcutaneous entry? And hydroxyaluminium actually is fixed to go, and however, we think that we had to test other relevance and the conventional medications, actually, and it is similar to, and everybody is aware about the, and we use the ointments and the adults and A2PXG, and actually the clinical trials demonstrated the efficiency as a content of our vaccines, and actually it was important for us to pick the regime of immunotherapy, actually, and it was really important for us, to pick the right period of having the vaccine administered. And it was important in terms of ambrosia plant, which is one of the leading ones for the and it was really important to get the protein. I will actually have the analysis, and we express the protein in the system, and we demonstrated. So we actually. And actually, the natural protein was not glycolized, and actually, there's a good generation of protein. So, and the purity is 95% and more. And that actually demonstrated the efficiency of our protein, which is and this actually refers to isotypes of class G. And we would work with immune globulin, however that data was not corrected with the real theoretical effect, and we took the EHS, and so that was a standalone work, and this was something that we worked on really, thoroughly. So this is as if you go to the allergy expert, and then you make the allergic agents, and then they watch the papilla. If it exceeds 3 millimeters in diameter, then it refers how how sensitive you are. And those experiments, they were assessed with the invasive methods, and we looked at the inflammatory reactions, and we would have the allergic provocation after a scene. And in accordance with those two results, it was demonstrated that the most effective in order for us to achieve the objective, it was adjective A51. So over here you could see the final formula. So it's 22 microgram per course. So we have adjacent therapy. So this is something that I mentioned about. So we actually managed to have good results, and in terms of ultra short regime, and whoever is interested, please, you may have a look at it. And later on, so one more important aspect: what the problem is. The patient who is allergic during the allergic season, has an appointment in doctors, and the doctor, can't just say that during the allergic times, there's not going to be any therapy, and the allergy may be getting even worse. And then the patient is given a symptom-based treatment, and then he said, come back during winter, and then the patient is doing what? During winter, he or she forgets about coming to the patient, and then, this is something that is getting cycled. So by doing so, the treatment is not received, and then it's not just the rhinitis, it's conjunctivitis, and then the asthma, and then it is very complex to treat. And in our study, we looked at the allergic season. For instance, we would disperse the allergens on the camera, and we would have, and this actually,
And this was actually the modeled system of the season of allergy. And plus, and that was, much more effective than the commercial similar proposals. So where we are. So currently, the level of biotech preparedness is really high. And at the moment, we have completed the pre-trials, preclinical trials, and the safety was proved. And we have patents in 80 countries. So what do we need to do next? It's important actually to make sure that the innovations are not left in the labs, and we believe in our products, and we're expecting the program from the Ministry of Healthcare, and perhaps by the end of this year we will be able to make sure that we achieve the 7th stage of the technology distribution. So it's going to be validated, and we also going to be having the first phases of the clinical studies. So this is something that we want to complete before 2026. I just wanted to thank. I'm completing my presentation. I would like to thank our partners who have provided us support, and actually different types of support, and sometimes some of them provide financial support to us, some of them provide support in terms of biotech, and say DNT-Vax and HAVA. And you could see our personnel members who have participated to the process. And so thank you so much for your attention, and goodbye. Thank you. So the next presentation, Ms. Kirichenko, Anastasia Sergeyevna Kirichenko. So the floor is yours, madam, please. Some distinguished colleagues, good evening. I would like, before I start, I would like to congratulate the National Center for Biotech with the anniversary, and wish you to have great health and all the best. And the topic of my presentation has to do with transfer of know-how, and today, during a couple of plenary deliveries, we've been mentioning about this, and it is important actually to have the applied science not to be left in the lab, or not to be left in the lab. We need to make sure that it's something that becomes a part of the clinical practice. And the modern practice demonstrate that there's a considerable share both of original and analog, have to do with the innovations, and pharmaceutical companies are interested in having original medications. However, a limited number of companies do have their own and this has to do with actually this is something that we have to adhere to at the early stage of development. So the requirements in terms of the quality of the developments, the state standards, and that have to do with all of the stages of the scientific development. So we are the
chains of the life cycle of the medication. So this is the foundation of it. We can't just get stuck. We have to transform and reflect the needs of the industry and market. And actually, it's important actually to get together with business, come closer to the business, because all of the rest of the stages, that include introduction of the medications, have to do with business. And over here we have to deal with lots of issues like PPPs, for instance, while implementing different projects, including preclinical trials and clinical trials, of course. The matter is that often, currently, what is happening is that the scientific center, is delivering some medication, and closer down to the level of getting a patent, and then there's an understanding that the pharmaceutical company is going to be having a response. This is going to be translated over to the production facility, and the product is going to be produced. But the other thing is happening actually indeed. So the results get scrutinized by the pharmaceutical companies, and one of the most frequently met is the method of the labs and which are not scaled up, and actually the pharmaceutical companies will need to develop the molecule from scratch, all the way down. And so let us talk about the problems overall in terms of interaction between science and business and the solutions that we may find. So number one is harmonization of approach in terms of identifying the results and quality of the work. Harmonization of the procedure of implementing certain stages of the projects while creating the modules aimed at transfer to include preclinical and preclinical trials and internships. So training for personnel members is a standalone issue. Harmonizing the approaches to project management, legal, bureaucratic issues related to intellectual property. So, looking at all of these issues, those could be divided into two parts, and should the first part be more related to some changes that may be happening inside the lab or scientific facility. The second part has more to do with administrative measures and the management measures overall. So how this issue is being settled in scientific facilities in Russia is that they create the Center of Medical Technologies Transfer, and this center has been represented by the regulator, and the expertise of medical tools under the Ministry of Healthcare is aimed at ensuring favorable conditions to transform ideas into the product that is demanded at the market. So the objective is to ensure the maximum efficiency and the transfer technology at all the stages to start to include all of the stages comprised of licensing. So the slide depicts more detailed information fostering that. So what instruments do we have in order to put together business and science, and the way we envisage it at the moment that are deemed to be more effective? So cutting the distance by introduction, so business instruments in daily practice, and quality management systems oriented for risk analysis, project management elements, having additional services, analysis of needs of pharmaceutical companies, harmonization of processes happening within the labs and within the units with GXP requirements, and the expansion of the list of accreditations with the maximizing the internal pharmaceutical production processes with the standards. And it's important to mention about the local instruments of putting those parts together. So number one is expansion in terms of needs of experimental data by creating adequate and valid model for evidence-based practice, introduction of adequate design of experiments, and thanks to the listed criteria. The criteria for experimental groups and getting rid of the errors and elimination of subjectivism and so on. And should actually, when we approach modeling of some disease, in order to find the proofs of efficiency of the medication, it is actually important to have the adequate model for the experiment. So the adequate design of the experiment. So firstly, we speak about the equipment that we use for the studies. And the equipment is to be validated, is to be fitted in terms of the maintenance details. And I was happy to see amongst the partners of the conference the representatives from some of the equipment producing companies and the adequate design of experiment. Again, we're going back to the efficiency and the kinetic studies of biomedical cellular products when you need to have the visualization with the fluorescent and luminescent sources of light and CT. And certainly we should be talking about xenograph models, solid tumors and liquid tumors, in order to have the efficiency of the medications proved, like the CAR-T. And over here you could see the pros and cons of fluorescent and luminescent imaging, and this is needed in order to have the valid results received. As a reference, you could see the pictures made during the real experiments of solid and liquid tumors while testing products of CAR-T. And couple of examples of introduction of innovations. So the materials were provided with the agreement of 2 labs who participated in the actual experiments. Pokrovsky's lab, Russian University of People's Friendship, the colleagues are dealing very actively with the cancerous diseases and testing different prospective molecules together with the pharmaceutical companies, and they have arranged and managed their operations in accordance with the principles that we have mentioned about during the presentation. And they have some culture developed works in vivo. And over here you could see the major anti-cancerous agent related studies in molecular oncology and characterization of tumor models. It was very, very active lately. They've partnered with different pharmaceutical companies. And another reference from Kazan State University, Mr. Usachov chairs the lab of structural analysis of biomolecules, and they are looking for antibiotics resistancy. And in fact, the objective of studies is liposome, and they've succeeded a lot, both in discoveries in terms of mechanisms of resistancy, as well as reaction to different pathogenic microorganisms, as well as they have developed the cell-free systems of translation. So this is one of the applications that has to do with the new T systems. So
The conclusion is on the screen, so allow me not to read it, and I would like to keep up and say that we're going to be hosting an event in April. We used to be having this April, and next year is going to be the same technological workshop, R&D, that puts together the developers with the pharmaceutical companies and investment funds, where you will have a chance to discuss all of these issues. So thank you so much for organizing such a nice conference, for your invitation, for a chance to talk to you. And I would like to thank the sponsor, a company called Advina. So this is the first time for me in Kazakhstan. I'm admired, nice and warm people, and I will try to come back. Thank you so much for your attention, dear colleagues. The next report is going to be made by Mr. Valerio Kozak of 4D, Proteomics and Metabolomics.
Right, all right. Thank you so much. Yeah, good afternoon, everyone. I'm so sorry. I really tried to make a talk in Russian, but I failed miserably, so it will have to be in English. And I do represent today Broken Daltonics, and we will be talking about a timsTOF system that is addressing different challenges of proteomics and metabolomics in the current landscape. So let me give you a bit of a context. Of course, systems biology is important, and just genomics doesn't lead us anywhere, because we do have a lot of biochemical processes that are influenced by foods, medicines, environmental exposures, different lifestyles, etc. And that, in turn, influences our molecular biology on proteomics and on metabolomics level. And this way, analytical techniques that are planning to address that are kind of in a pickle, because we are having a tie between high throughput analysis, in-depth analysis, and high sensitivity. Essentially, whatever is high throughput is typically not in-depth or not highly sensitive. That in turn is being chased by a dragon of data analysis, and our data are really complex and really big. And that is prompted by demands of reliable quantitation, dynamic range that needs to be broad, missing values problem, ID numbers need to be high, and of course, as any analytical technique, we need robustness, reproducibility. Well, we are aiming to address that with 4D omics, what it actually is. So in classical proteomics, we do have for our analytes such a parameter as retention time and the LC separation level. We have MS1 data that measure accurate mass of the compound, and of course we have the MSMS fingerprint that gives us structural information from fragment ions pattern. In Bruker, we have inserted into the TOF system a TIMS cartridge, which stands for trap time mobility. And now the value of collisional cross-section, or CCS in short, that is measured in the timsTOF system is the fourth dimension for identification. And it really helps not only to identify the compounds, but also to separate and space and time focus them within the TIMS cartridge. So principles of operation is really easy. We do have our analytes coming in the gas phase from the ESI source, and they are being carried by the N2 gas further in the TIMS cartridge, but they are in turn are being stopped by the electrical potential that is applied on the TIMS cartridge. And this way, at a certain point, they would reach an equilibrium with the larger molecules, and their cross-section would be first and would be further in the cartridge, and meaning they would be eluted from the cartridge first and travel further through quadrupole to the TOF. Not only that, our cartridge is actually a dual one, meaning that while we're doing the analysis in the second cartridge, the LC flow doesn't stop, and in the first cartridge we are having accumulation of the ions coming off the plume, and further on they're being transferred in a sequential manner to the second cartridge. And that, in turn, is synchronized with the operation of the quadrupole. And with that, we've coined another term of parallel accumulation, serial fragmentation, also known for short as PASIF. And that synchronization allows us to do really incredible things. So just to bottom line that, the dual TIMS analyzer allows us to achieve 100% duty cycle. We never lose ions coming off the LC, and most importantly, unlike some other analytical techniques measuring ironability, here CCS is actually measured and not working as a CCS filter. And that all is coming at no additional cost whatsoever, because in fact our chromatography is a pretty long process. 7 seconds is really, really long. And ironability separation, several events can fit in that width of the chromatographic peak. Further on, we have our quadrupole filter, where mass selection is happening even faster, and our TOF is really, really, really fast. And essentially, we're getting some sort of the nesting doll situation where we don't really sacrifice anything in order to get that semi-orthogonal separation built in. Oops, sorry for that. And yes, we do have a range of instruments that ask you to solve different tasks. So the initial one was a TIMSTOF Pro that was introduced quite some time ago, but further on we grew our portfolio through TIMSTOF HT to TIMSTOF Ultra 2, and they are all getting improvements on both hardware and software level. And now, enough of the boring details, let's move to the examples, what we can do with that system and what our customers are doing with the system. Starting with metabolomics, I would like to focus on the benefits of the 4D proteomics for the lipidomics workflow. Well, first of all, we are able to separate isobaric lipids, and you probably know if you work in metabolomics field that that is indeed really important in order to achieve good quantitation and in order to achieve good identification as well. So those lipids can be identified confidently using MS2 fragmentation, but we can confirm that annotation based on their CCS value that is measured in TIMS. The first and the fourth steps would be to validate the finding using Kedrick mass defect plots from the isotopic distribution, and then we identify biomarkers comparing contrast to the disease cases, for instance. And an actual real-life example of how it works could be shown on those two lipids who are virtually isobaric. So the mass is 7-fold for both of them, but they belong to different lipid classes. And in a 3D experiment that you see here now, they are co-eluting on the LC separation, and therefore quadrupole won't be able to resolve them either, and we are getting fragmentation spectra that are essentially chimeric. So they are mixed, and confident identification, not to mention quantitation, is not possible anymore. However, if we introduce the ionability dimension, those lipids are actually resolved on the ionability dimension as, do we have a, yeah, as we can see here. And we, therefore, can send them to quadrupole separately in time, and this way we're getting two different fragment spectra for those lipids, and we can quantify and identify them confidently. And that, of course, can be also done on the high speeds of the TOF operations, because, of course, with those extra dimensions of separation, we are decreasing the sample complexity dramatically. Therefore, we can afford shorter gradients. And in fact, in the blink of an eye, only in 4-minute gradients, we are able to characterize brain, cells, stool, liver, and sclera specimens in terms of the lipid content. And on average, we were finding more than 500 different lipid classes for all those real-life samples. And given those are clinical samples, that 4-minute time is actually quite crucial to manage with the cohort's volume. Oh, what did I do? That's not mine. All right, I'll click quickly through it. And And that speed can also be achieved on proteomic samples as well. So here you can see in terms of HT, we did a ring study across 11 laboratories in the European area, and we have injected 10 replicates of 200 nanograms of K562 cell digest, and the gradient was only 5-minute runtime. And we were confidently identifying over 7,000 proteins per replicate and quantifying a little bit under 6,000 proteins per replicate. So the depth of analysis only in 5 minutes is really amazing. And indeed, that throughput shows in our satisfied customers. So here, actually, you can see tip racks, and those tips are used in that EVA subsystem, with each tip being one sample. So 96 tips, that's the height of the tower with our customers, and indeed the throughput was going on 24/7 non-stop. So the robustness of the system is really great as well, which matters in clinical research. But the same system in terms of HT. HT stands for high throughput, but not just that. It can also go deep into the analysis. So further on, we have investigated how we can go deeper with the same system using a very long gradient, relatively long gradient for terms of operation, and a rather long chromatographic column, 60 centimeters. We were able to detect over 10,000 proteins from cultured human cell lines with a single shot proteome. So there were not summations of replicates, no MBR was employed, and similarly impressive results were achieved from plasma and serum, averaging over 8,000 and 4,000 unique proteins respectively. That was done on the DIA mode of operation, and here we have employed a new technique that allows us to follow the ion cloud precisely, therefore not wasting the cycle time. Another application that is kind of blooming right now is, of course, single-cell operation, and here we're moving to another class of instruments that are offering high sensitivity. Speaking of single-cell research, typically we're dealing with tissues, let's say cancerous tissues, and if we just blend them together, we are losing the spatial resolution and cell selectivity while the tissue is rather heterogeneous. So here we have different cell types, different stages, different clonal cells, and we are not able to retain that information to gain more insights about the biology. But this way we're moving to single cell and optimize every step of that process. We are able to retain the spatial resolution and cell selectivity and also dive deeper into the tumor tissue, for instance. However, that comes with needs and requirements, of course. We absolutely need an ultimate sensitivity because the amount of material is really, really low. But also we really need high throughput because that is a lot of cells and we need to address them all. And the robustness of the system matters a lot since we are only having single shot per cell, so we can't afford missing out values. And this way, we are able to actually tackle different types of cells. They're still single cells ranging from oocyte to very, very small lymphocyte, 20 picograms of material, and different blood cells can also be addressed. Sorry. That was not supposed to happen, can also be addressed without the cell type specific sorting. We're gaining easily over 2,000 protein group identifications, but more importantly, we are able to separate those cells on VCA without prior information from the sorting stage. Another application that is already mentioned during this session, the immunopeptidomics, is also very, very important currently in the biomedical research because indeed each cell presents peptides on its surface, antigen peptides, that are being screened by the immune system, specifically by T cells. And the identification of pathogenic, mutated, or otherwise damaged cells is happening by those antigens, and the immune response is triggered. Therefore, we are paving the way to translational research with understanding the mechanism of the immune system and specific responses, and that would help us not only understand the disease pathogenesis and development, but also to find new immunotherapies against, for instance, cancer or infectious diseases. So that research is quite important, but it also comes with challenges. Those peptides are in really, really low abundance and are really difficult to generate. So a lot of prep work is needed in culturing cells and treating them. Therefore, we want to reduce the sample consumption as much as possible. Oh, Jesus Christ. So sorry. I broke everything again. Right, so we want to reduce the sample consumption as much as possible, and we want to limit our preparation for the actual measurement stage from culturing the cells. And that's exactly what was done by the company Althair Bio, and they have started with just 25 milligrams of primary humoral tissue and were able to decrease the amount of injected material without losing the quality of the analysis by 3/4 compared to the previous technology. Another topic that I would like to present here is, of course, a PTM perspective, because TIMS really helps with allobaric peptide separation, meaning it also does help with phosphopeptides. So we're able to decrease the number of car-isolated features quite dramatically, but also we separate them and we can see that they are being addressed separately in the quadruple and TOF operation. So here is the example of a standard triptych peptide, actually 4 sequences here, and they all are being separated on the TIMS dimension and confidently identified as phosphoisomers with the localization of the phosphomodification. And another application of PTMs is, of course, glyco. It is rather difficult to analyze glycosylated peptides, and here I just want to demonstrate the recent workflow published on a timsTOF system. What I would love to highlight here is not just the workflow that we can do, but also the fact that this workflow is being supported by the software solutions for glyco. And I really wanted to talk about metaproteomics as well, because it's kind of a big thing right now as well, given the volume and the low abundance of the species in the proteome and metaproteome. But if you would like to talk a little bit more about it, I would be hanging out right outside of this room and would be happy to chat about that or anything else of that matter. But that is just the tip of the iceberg we scraped today. So here we are indeed growing quite fast in the number of publications about the timsTOF, and those were just selected applications that we can do, and indeed we'll be happy to chat about that as well. So thank you so much, and yeah, I guess I don't have any time for questions, but we can talk later. Thank you.
Okay, thank you very much for your presentation. И мы завершаем последним докладом.
And here comes the last presentation. Zhamankulova Karlugash is going to be talking about the biological, biotechnical medicines. Please, the floor is yours. Good afternoon, dear colleagues. And I represent Kazakh and I want to show you the farm market in terms of biological, pharmacological. And you could see the classification of biological and biotechnological medications, which is the biotech is really and the classification would be enlarged actually. And you could see immune therapy and the advanced therapy, and there's a standalone blood products. So all of the groups have pretty similar titles, but they are different in terms of technology, effects, and so on. And I want to demonstrate the state regulation of pharmaceuticals in Kazakhstan. So the first column contains information in regards to legal acts on safety and quality of biological substances, starting from the Code of Healthcare and the law of healthcare services in Kazakhstan. And in fact, there are more legal acts in Kazakhstan, which is over 100, and actually international legal acts do have lots of lists of provided by EMA, FDA, and so on. And all of those legal acts are aimed at safety and quality of medications. Analysis of the state register as of April demonstrates that in terms of turnover, we have over 7,000 different medications, and over 5% is biological and biotechnological substances. And to be precise, out of 7 groups, we have 4 positions of medications, so biological, so immune biological, immune and medical products. So distribution of biological products are aimed at the market look the same, and we have the largest number of products in blood and bloodstream, and then antimicrobe products, and in 5 classes we have biological products registered. And over here, I wanted to demonstrate that the largest number of biological medical products are made as injections, so suspenses, solutions for injections. And this slide demonstrates the structure of producing countries who have registered their products and medications in Kazakhstan. As this slide demonstrates, overall pharmaceutical market of Kazakhstan is dependent. So 15% of medications are made locally. The rest, over 80% of medications, are imported, delivered from abroad. And in bio products, the picture is the same. So out of local products, it's slightly more than 15 medications made in Kazakhstan. So you could see domestic biological products, and out of industry, biosimulator producers, we have 16 medications made. So we also have a local vaccine registered, made by the Institute of Special Infections. So this is how the picture looks like in terms of local products. And we wanted to mention about the scientific centers dealing with the biological and the biotechnical means. And as you have seen previously, some of the centers were dealing with lots of innovations and discoveries. And here comes the National Center for Biotechnology, the National Center for Dangerous Infections, and Institute of Biological Safety. And we also have antigen facility dealing with the biological product production, and there's also a scientific center for injection products, and so we also have anti-TB products and Institute of Genetics and Physiology and Microbiology and Virology Institute exists in Kazakhstan, Phytochem, and the Institute of Plants and Microbiology of Plants. And there are also other less prominent centers in Kazakhstan. We wanted to demonstrate that out of 5% from the overall biological products, more than 50% is purchased under the program of the guaranteed medical assistance at the state budget. So we've tried to demonstrate some SWOT analysis of the pharmaceutical market, so the strengths and weaknesses. We're limited in terms of time, so it goes without saying, and that you are aware about the problems that we have. And also we've listed the threats and the potential for delivery, for the development of the farm industry. So that's all that I wanted to deliver about, and I wish you to actually enter the market. And thank you so much to our last presenter and those who have survived until the end of this wonderful session. I'm going to be making a couple of announcements. At 1900, there's going to be 2 buses that will bring you over. So those who live in the hotel, please be aware that there's going to be gala dinner, and the second bus is purpose for those who are not living in the hotel, and you are invited for gala dinner. So the tickets that we have given you earlier is something that you need to have with you. So we won't be able to show you all of the labs, but we can start from the 3rd floor. And at 1900 we need to enter the buses. Otherwise it's going to be traffic jams, so it won't be convenient for us to move around. So those who want to have a tour, let's start with the 3rd floor. Thank you and goodbye.